hands. We ask for your leading, for your guidance, for your direction. Every aspect of this conference for today, let it be guided and ordered by you. We ask for your wisdom and direction in every aspect. Father, in the mighty name of Jesus Christ, keep each and every one of us. Let come to pass, we shall enjoy your leading and your guidance. We thank you and we give you praise. For in the mighty name of Jesus Christ, we pray and believe.
Okay. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Ar-Rahmanir-Rahim. Maliki yawmiddin. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal ladzina an'amta 'alaihim ghairil maghdubi 'alaihim waladdallin. Amin. Bismillahirrahmanirrahim. Qul a'udzu birabbin nas, malikin nas, ilahin nas, min syarril waswasil khannas, alladzi waswisu fi sudurin nas minal jinnati nas. Bismillahirrahmanirrahim. Tuanze vyema na tumalize vyema. Amen.
Thank you very much, Madam Hamida and Mr. Kenneth Waitito. The Board of Directors, KIPRE, the members of the International Advisory Board, the organizers of the 20th KIPRE International Scientific Conference, all our delegates, our partners, collaborators, good morning. I want to take this opportunity to invite you to today's sessions. This is our third and our last day since we started this International Scientific Conference on Wednesday. And I want to sincerely thank you because you have persevered and you have ensured that you have been patient throughout the 3 days. This is going to be the last day. And before I invite our speaker, I wanted to recognize two individuals in a very special way. Professor Mwenda, who unfortunately is not alive but is on the way, and Professor Thomas Kariuki, who is going to be our speaker. I want to recognize those two because they are former directors of KIPRE, and they have continued to support the institution. And to me, it is a challenge to all of us that after this conference, go back to that primary school that you went to. Go back to that secondary school that you went to. Go back to that university that you went to and do something for those institutions, because it's good to give back to where you came from. So I'm privileged to introduce our speaker today, Professor Thomas Kariuki. And Professor Thomas Kariuki is a globally recognized immunologist, a primate scientist and science leader, currently serving as the founding director and chief executive officer of the Science for Africa Foundation, SFA, a Pan-African organization established in 2021 to support and promote science and innovation across the continent. He is a passionate advocate for research on poverty-related diseases, and he has played a pivotal role in mobilizing hundreds of millions of dollars in funding from global donors and African governments. Professor Kariuki's leadership is driving the creation of strategic global partnerships and a sustainable science funding ecosystem in Africa. Prior to leading the Science for Africa Foundation, he served as director of programs at the African Academy of Sciences between 2015 and 2020, and as a director and senior research scientist at the Institute of Primate Research, currently KIPRE, which is a WHO collaborating center for tropical diseases and human reproduction. Professor Kariuki has received numerous honors, including the Order of Grand Warrior of Kenya, OGW, and holds fellowships and honorary positions with the African Academy of Sciences and the Liverpool School of Tropical Medicine. He has served on global boards and committees such as CEPI, WHO, and the Federation of African Immunological Societies, making him a leading voice in African and global science. It's now my privilege to invite Professor Kariuki to talk to us. Karibu, Daktari.
Morning, everyone. I've come a little bit on a rush because I wasn't quite sure how to get here sometimes. I don't know about you, but every time you come around here and you have to do a lot of roundabouts, and then sometimes you go up and then you have to come back down. But I'm delighted to be here. I was invited by some ladies I'm seeing here. And because I've worked with them for many, many years, and because I handed over to my immunology lab to them, it's very, very difficult to say no, and it's very difficult to say to Peter. Let me spend some time with you. First of all, thank you for the introduction, Peter. I forget sometimes about my own introductions, which is always a good thing sometimes. But let me say something about myself, a little bit about me, but I'm not getting into too many things because I think this meeting is about actually looking at what happens, what we should be doing, the next generation of scientists. And I spent almost 20 years at the Institute of Climate Research then, Kipre then. I built my early career there. And I believe that we established with, I'm looking at you, Marcy and Ruth and of course Lucy and all the team, but I believe we were able to establish a vibrant immunology research group there, which I think have very much survived me since I left in I forget even when I left, Peter. So institutional leadership and building R&D infrastructure has been my passion. And I think this is something I have continued long after I left Kipler. And I did that because, of course, all of us struggle to secure grants from across the world. So I thought maybe I could step into the other direction of things rather than being a grant recipient, to start to explore whether we can, the organizations that I was going to get into, like the African Academy of Sciences and now the Science for Africa Foundation, whether we can become a grant funding organization. And that's about building for Africa, building for Kenya and the countries, for targeting global influence. And our contribution to science and innovation globally has been my passion since then. I was looking at the advisory board meeting earlier this week, and Steve Shapiro, who is here, had some great photos, and I couldn't find some great photos myself, but I found this one, which reminded me of some of the people I have worked at Kipler. And I can recognize some faces here. Oh, no, maybe, well, Dr. Shai must be here somewhere. Yes, so I think it's good to see some of those familiar faces, and a great reminder of what a great group that I was working over the years. I think for anybody who is aspiring to be a science leader, I think I've learned along the way some of the things one has to have. You've got to have some ambition. And ambition, not over-ambitious, but it's good to have ambition. It's something I learned in leading institutions is that innovation is great. You have to keep innovating organizations, but I think reinventing the wheel sometimes can also get you off the rails. You have to make difficult decisions. I have had to make some difficult decisions, including where I want to leave an organization, and unfortunately, who to offload from an organization and all of that. It's interesting when you work in public service or public organizations or parastatals like Kipre, as opposed to work in an NGO like the one I'm working in as an international organization. A lot of lessons one learns, and transferring those skills is interesting in itself. I'm driven by science to impact. One of the things we all struggle as scientists is what do you do when you do all that great immunology, when you do all that biochemistry, all the molecular biology and all of that, then what? And the questions when you leave the lab is always the question about, is a publication the end of it all? And I think this is the challenges we have from our society, particularly from Africa. I think the template that has been around for too long is about a template that actually we sort of imported from out there. And that is something that I would like to challenge over all of you, particularly when I talk to academicians in universities. I ask that question about, is it just publications about whether you're going to get a promotion, or is there something else that is going to move beyond that publication? Because we need innovations to come out of our academia, or from research institutions like KIPRE and all the other institutions we have. I think I found out in KIPRE and elsewhere that I have worked that we have a huge, brilliant talent of young scientists, and I think Kenya and Africa are in very good hands if we can harness that talent that we have. 30 plus years ago, every time I went into meetings, there was always a question about, if we bring a program in Africa, do you have the best talent to deliver? Do you have people who can do discovery sciences? Why don't you stick to, for example, just doing clinical trials in Africa? Why do you think you should get into discovery? And yet the investment for discovery sciences can be huge. You need structural biology, you need sequencing, you need all of that. But I think we're in a much, much better place here in Kenya and across the continent to do cutting-edge science. And again, that is something that I found I would be very happy to continue to support as we do that. And then governance, of course. Governance is accountability. How do we get assurances? How do we actually keep making sure that accountability, transparency, nobody invests in an organization that actually have challenges when it comes to governance issues. Let me introduce our organization, a little bit about our organization. I'm struggling here, Peter, with this, but it's okay. So the Science for Africa Foundation, some people, I think we are still a startup because we started this journey of building a Pan-African organization 4 years ago. However, what we did was to transfer a platform that was hosted, or rather to recreate, or whatever the right word is, a platform that actually was doing the kind of work we have continued. So we are a non-profit. We are a public charity organization. We are not a private organization. And our mission is to support, to strengthen, and to promote science and innovation in Africa. Our strategy for the period of 2022 to 2027 is around those three science thematic areas. Health, securing global public health security, very big things as defined. Climate and environment, that's about driving global climate change adaptation, mitigation, resilience. We know that Africa is the most negatively impacted by climate change, and we are unprepared, have been unprepared for this. And this is connected also with agriculture, how we can advance to secure food security, or protect our biodiversity, how all of that can be interconnected. I put that triangle because everything now is connected. I think the vertical thinking that has been trained going through education, that you can bucket yourself in this area and others bucket themselves in another area, it doesn't work very well now. And I think all of us have to think about one system, one health, one approach to how we do science and innovation. And the reason for that is because changing climate patterns that have a direct effect on food, which in turn, food and nutrition, that means it impacts health outcomes. So a household that cannot actually produce sufficient food, and that is because there is climate challenges, means that there is no health well-being within that family, within individuals. That's why, therefore, thinking around the interconnectedness of all three areas has been something we have been, at the organizations, a priority for us to support. What we frame our core businesses in 4 words. So as a Pan-African organization, it's a huge continent. The more you listen, the more you learn about where the priorities are. And we do not want to be presumptuous to say that we know what the priorities are. So we listen, we convene. And that means we are looking for evidence-based prioritization of challenges and gaps that are solvable through R&D. Now, not every challenge in the world is going to be solved through R&D approaches. But we commit to that if there are gaps that can be solved through R&D support, we strive to do that. That leads us to do the design of programs, because those design means we need fit-for-purpose programs that can then address some of those priorities. So it's the models of design, it is structures, programs, it is systems, it's policies that need to be implemented for us to do optimum delivery. And then, of course, most people, they know us as a funding, grant-making organization. Although we don't like that because we do a lot more than just a funding organization. And the reason for that is that we don't want to be seen as a traditional organization where I think all of us, you are familiar with the model. You apply, you get a grant, you sit back, you do your work, you make a report at the end of it, and that's what is required of you. But we look at our approach to funding and grant making as a collaboration, as a partnership. And so we don't just give the grant and then we sit back and we wait for a report. We actually make visits to institutions. We get to know each other continuously. We keep informing each other, information flow, knowledge sharing, learning among each other, even as the organization deliver on what is required of them based on the grants that have been provided. So sustainable structuring and financing of impactful R&D initiatives. And that's long-term vision. And then the fourth area is accountability. Accountability is about oversight. We have to be transparent in monitoring what it is that we are providing. That means we have to use best-of-class metrics around this monitoring, because we have assurance requirements to those who are our partners, assurance to our funders globally who provide us with the resources that we put into a pot of money that then goes out. And that accountability to our partners is not just our funders, it's also our partnerships with government, it's partnerships with institutions. That's the assurance accountability that we really put as a priority for our work. I'm stuck. There. Okay, there we go. So we have 4 areas here, again, where we want to see success. Success in leadership and excellence in science. Definition of excellence, all of us can have our own definition of what excellence is. So we look at that One Health approach. I keep emphasizing on that, for whether it's infectious diseases or NCDs or One Health consortia, how we build them. We have recently focused on mental health. Why? This is the youngest continent, but you look at the numbers for depression, anxiety, or suicide harm, self-harm, all of that. I think we are suffering from a huge crisis of mental health. So we are providing grants now on mental health, and this is through a consortia model, and I'll say about how we build through those consortia. Social sciences are neglected. I hope there are social scientists in this area, because that actually has been a challenge to build around social sciences and humanities. And that we know when we do social sciences, we're talking about communities. Communities are the one where the problems start, and solutions must be embraced by the communities. Some of you may know Karistas Juma, or the late. He's one of the most celebrated scientists wherever he worked across the world. So in honor of Karistas Juma, we started a fellowship called CJ. We worked with the Gates Foundation to start putting together these fellowships. And they're really about developing of leadership by providing fellowships. So this is about training, mentorship, leadership development, and amplifying science voices across the continent. Our second portfolio is how science innovation, transition, and entrepreneurship is done. Again, we are all used to getting support for a proof of concept. We provide grants, for example, for people to do 18 months to 24 months of a proof of concept. Those sometimes are what we might call seed grants. Seed grants could be, say, for example, around $100,000 for somebody to start over 18 months to start to do the proof of concept. But what we are really looking for is the next phase of that. What next after those 2 years? The transition to scale. And that's one of the weaknesses we have in Africa. And I keep emphasizing that point, because even universities, who of course are supposed to be generators of all the knowledge we have, that knowledge is getting stuck, because at the end of the day, it's not ending up as innovations that actually could end up with the society challenges, address society challenges that we have. So transition to scaling is an area where we are determined to continue to support institutions and individuals who are thinking there around that. The third area of our portfolio is around science solutions for emerging global priorities. And this is where we are looking at epidemics, clinical research and clinical trials, climate, health and agriculture, and biodiversity. Those are all emerging global priorities, particularly around pandemic sciences. So we learned a lot from COVID-19. We learned just how we got to be at the end of the queue, whether it is getting access to vaccines or diagnostics. We learned that if we were to then build self-sufficiency, we need to start thinking now and build that level of preparedness, response work in very broad partnerships. So we have awarded 7 large consortia, very much focused on pandemic sciences. I don't know whether pandemic sciences is something you can teach at school anymore, but I think it's many, many things that come into that. Consortia focus on, for example, vaccinology, biology, so they can look for everything from genotype to phenotype, public health, clinical trials and clinical research, social sciences. All these are large consortia that actually are focused on how we can then get that preparedness. Hopefully, there won't be another pandemic, but that means that is just the focus. The focus is infectious diseases, but then, of course, it brings all of that preparedness and response if, I guess some people would say not if, it's when the next one might unfortunately happen. Then the other area is to do all of this is a portfolio around strengthening science ecosystems. This is about institutional capacity strengthening. Good science comes from ecosystems that are actually strong enough. They have to be conducive. They have to be research focused. They have to be having the tools to enable them to strengthen their offices. So we have developed standards. Good financial grants practice is one of the standards, and KIPRA is one that has been a level of GFGP that actually then presents the opportunity for KIPRA to continue building their status as an emerging, well, a well-known but more accelerating the work that KIPRA does through having the standards and eventually being accredited to some of these standards. And last week we launched a second standard, a Good Research Management Practice Standard, GRMP. And that again is to look at how we can support institutions with everything from how ethics around governance issues, all those areas that are required around all the science that needs to happen. So, and then of course there is the Open Research Africa Publishing. We have a platform. This is about open science, open research, broadly speaking. One of the challenges we all suffer is, yes, you want your publication in Nature or Science, at what cost? And in my career, I struggled to sitting with, say, a government official or a minister in a room who have ever asked whether you have an article in Nature or Science to help them to make a decision, because that's not how governments work. You have to then, yes, you can publish in the best journals. That is important. That is part of it, is, we do as scientists. But again, the question is, how do you move from data? You synthesize that, the evidence, you then present it to actually policymakers so that they can use that evidence to make decisions. Because decision making is about having that kind of approach. And what we have done here is to build a platform that is open access, open publication. You don't, the amount of money you pay to get published in some of these, let me call them global journals, probably if you look at, you have to pay $13,000, $12,000 to actually get published. That is like the salary of a student working in your own lab for 6 months or for 1 year. And the question we have to keep asking myself, is that really the approach? Are we just adopting a template from outside the continent? And to what extent do we want to do that, as opposed to more innovative, more metrics? I have always disliked these things about impact factors. What is impact factors? Impact to who? Impact for what? And I think universities, in my view, I would challenge them because they are stuck in time about this impact factors issue. And I think this is something that I hope that we continue to think as African scientists, African ecosystem, about how we can do such more innovative ways, but affordable ways to publish, to get access to data, synthesize evidence, and that can be more transformative. Now, across all of these things, this area, we have partnership resource mobilization, we have science communication. Science communication is critical. Again, scientists, please, I know it's easy to publish with all the jargon with our science. As a mineralogist, of course, it's great to do that. We have to do that. But when you communicate, you have to communicate that something that is understandable. None of the jargon of science if you want to engage with policymakers. Now, science communication has become important also because of the geopolitics. We all know that we have this, the shift that we are seeing in geopolitics. How do we make sure that science remains something that is embraced across the society, across governments, embraced in decision making, and we don't get swayed by this transient, hopefully there will be transient geopolitical shifts. Community engagement with science is about working in communities. Policy and knowledge transition is something that we look at across all the areas we support, and we then collectively then have to do management and learning from that. Now, when we put out requests for proposals, we ask consortia to come together. We usually get, so the last one when we put out consortia, approach of a hub and spoke model. The hub is the lead organization. The spoke are partners that come together. We get 150 consortia that wants to apply from across the continent. It's a Pan-African organization. Money is very, very scarce, so it's very, very competitive. What I find sometimes with all this, I mean, we also have models where we give individual investigator models. Last year, we started population cohort model where there are more than 60 cohorts across the continent. Some are small, some are medium-sized. One of the things, again, that we see in Africa is the fragmentation of what is happening in every corner of the continent. So 60 of them, some of them very duplicative about what they are doing. So what we are trying to do through a secretariat we have developed to make sure that we have this harmonization, standardization of what these cohorts are doing, so that we can then embed some great science questions in those cohorts. We have started on that as one of the model, and then we do networks, bringing together some of the existing networks, not necessarily because we are funding them, but because they exist and we can actually learn from each other and support them in their next phase of development. So I would say, for example, thematic network model is one, some of you may be familiar with the one called GISA, which is focused on genetics, immunology, but a lot of focus on genetics and genomics work. That is a Pan-African, actually even beyond Africa, where we then support that network. That probably is my, I have a lot of support to see whether that is going to move forward, because of course, as an immunologist, I want to maintain my connection with people doing immunology on the continent. Now, and I've already talked about some of these areas that are so important for us in terms of moving forward. So I think those are just for me. So just to summarize about what it is that we do. All of these areas is areas where some of the funding we have has gone into these areas. NCDs, clinical trials, vaccinology, neurosciences. For the first time, I think we did neurosciences. We have a consortium led out of Arakan University here. Neurosciences, some stuff around child brain development. Some of those areas that we have not been done in many of the African countries. We have endemic, we have data, we have pathogens biology, we are supporting AMR work, we are supporting women's health. I think there are 4 areas in, if I can remember, there are 4. So one of the areas around genetic infertility, not infertility based on, for example, the clinicians who would say that they have done everything trying to figure out why a couple has been difficult to have a conception. That is something we are looking or supporting for genetic infertility. I didn't know, but actually I was told there is even like a genetic infertility belt in Africa from central going to east to the south. It's an interesting question to try and understand why that is the case. So of course, we are developing a lot of genomics work across the continent. Genomics so that we can then get into this level of confidence that actually we can have precision medicine which can eventually be embedded in universal health coverage. And we can make the case with confidence. I have a lot of discussions going with some of the government agencies here in Kenya like NACOSTI, around this area of precision medicine. Agriculture, biodiversity. So I'm summarizing everything that we have been put into the support we are able to provide. It's discovery sciences, it's transitional sciences, and implementation and policy. So what is our experience after all of this? For me, 10 years of this space, 5 years since we started Science for Africa Foundation, what have we learned? First is money. There is a shortage of money. Across the continent, when you look at the status, that is a challenge. It is an unmet challenge, despite all of us collectively the best effort we make. But you look at where the money, well, North Africa is doing better than Central Africa, for obvious reasons. I think they have been building the infrastructure for many, many years. They have very strong partnerships within that region. South Africa is, of course, one of the leading countries in Africa when it comes to sort of attracting funding for R&D. Interesting, we have a lot of funding that is going to South African universities, which doesn't quite address the question around equity. And I think we are struggling a little bit with that. So we would want to see a little bit more of equity with regard to where the funding is. So UCT is one of the recipients of many of our grants. But I was looking at the data from UCT. Despite being the best is relative, university in Africa, I realized actually that most of the recruitment and fellowship they are training actually come from other countries. So those students, fellowships. So they are doing the approaches the right way, in my view, about how they are approaching this. Eastern Africa, not too bad. But I think the most important thing, the fundamental question is, how much money does Africa require to really make an impact? So what we have is, if you look at all these Pan-African 54 countries, this is data from the World Bank. And what they figure out is that around $11 billion is available for R&D work in Africa. The gap is 14 billion because we require something like $25 billion on an annual basis to make change in Africa. It's a lot of money. But this is a huge continent, 54 countries, so everybody should really be focusing on that 1% that was pronounced by the African Union to make change in their own countries, even before we scale across the continent. So our small contribution to all those billions is being successful in our fundraising across the continent, across the globe. Much of our funding is negotiations with philanthropic organizations. That is a great source of funding. So I would say out of sort of 15 large philanthropic, our partners, only two international governments, Canada and the UK, have put money into our pot of funding. The rest are from the big philanthropic organizations. One of the biggest is Welcome. Welcome, the Gates Foundation, more recently Mastercard Foundation, but you have a whole list of other philanthropic who actually, and this is the thing, the uniqueness about this is that they are not the one who are telling Science for Africa Foundation where to fund, who to fund. Essentially, they are putting a pot of money collectively and saying, here is the money, go out and give it out based on the priorities for the continent. Some people would say, oh, are they the ones defining the priorities? Frankly, I can tell you, I actually don't know the strategies of some of these philanthropic organizations because it's not my business. My business is to present my priorities and ask them whether they are willing to put funding into those areas because those are the priorities for the continent. It helps if we have full alignment with those funding global partners that we have. Welcome, for example, as an example, they have a strong focus on mental health. They have a strong focus on infectious diseases. They have a strong focus now on climate and health. Those are things we can say we are fully aligned in our priorities for the continent. Currently, we have 146 grants. Some of them are very large. And by large, I mean each of those consortia could be receiving between $5 and $7 million each. So very significant money that actually can really do some good work across because the consortia are not embedded in one country, it has to be partners from other countries. 40 countries, we have a large footprint. 90 institutions that have been recipient in this support across the continent, and we hope we can award up to $250 million by 2027. Remember, it's billions required. And so many, many players need to come to the scene to really make a change across the continent to make sure that science and innovation is central and it's part of economic development and making change. Here in Kenya, because we're in Kenya, I have to say something about Kenya because there are people from different organizations here. $15 million in the last few years, I think the last 3 years. That's going to more than 20 Kenyan institutions, PhDs, postdocs, strengthening ecosystem. KEMRI is one of the large recipients. KEMRI Kilifi, not KEMRI Central. KEMRI Kilifi, as sort of, some of you may come from there. It's a state of the art. It's as good as it gets in terms of some of the infrastructure available. And I know for sure for you, Peter, and the team, that is the aspiration, to actually position Kipra as one of those, the best, that can actually use the resources. Everything is in place. And I think for Kipra is just to that step up to the next level. That I know that is a vision. Aga Khan University, we want to see more funding going to public universities. But the biggest problem we have public universities, again, is the quality. The ideas are abound. Making a compelling application has been a challenge. And that because there are no research support offices, innovation offices are not there. Even doing good budgets is a problem, sort of assurances that require to have the level of confidence that an organization have the capabilities to manage $1 million. That has been a challenge with public universities. And again, we have been trying to support these universities to do that. Government engagement here in Kenya, again, I have a lot of discussions with the Ministry of Health. Foreign Affairs, we are now, the State Department for Science, Research and Innovation is now being housed. And Climate Change and Forestry Environment Department. Let me revisit this aspect around where we are failing. We are failing because there is a lot of potential candidates on the proof of concept side of things, which can be fed in this pipeline of discovery, transitional sciences, hopefully all of them getting to the marketplace. So if it's a vaccine candidate, it doesn't, of course, stop at the level of getting a patent. And by the way, patenting is very low. I was shocked to hear that there are less than, 200 patents here in Kenya. I mean, the big players globally, they register thousands of patents every year. But that's because, although we have KIPI, institutions themselves are not doing much patenting. And again, intellectual property, we need that capacity. Last year, I got, as an example, one of the consortia we are supporting is based in Mali. Mali actually have excellent places where you can do science. If you ignore all the reporting from CNN and BBC about a country that is, I don't know what and what. There are actually gems of centers of excellence in many countries, including DRC Congo, where in places like Bukavu, they can do great clinical trial in some of these countries. But the problem is, all this is generating 30 years or 40 years of doing a lot of work. There are a pipeline we could be feeding some of these candidates, but they are failing down in that middle. You can see there's not much happening in that middle, the transition area. And that's where we need to focus on. And that's what we want to do, because you won't get a vaccine discovered in Africa. And let me just say that we are doing great clinical trials, but the question is always about who actually made the discovery for that. Doesn't mean that, of course, we don't want to do clinical trials based on discoveries made outside the continent, but the question has always been around ownership. Who owns that candidate? Because it's not patented here, it is discovered somewhere else. What is our contribution globally? And this is why looking at institutions is to make sure that we actually are doing a lot of discovery work. I've been challenged about that, about to say, oh, discovery, Africa is not ready for discovery work. But the transition aspect, let's focus on that. So one of the exercises we're going to do is, based on all those 146 current grants that we are doing, we want to do a mapping of all the potential candidates, whether it's in vaccines, diagnostics, whatever it is. And then we see how we can support this transition phase in the middle to get from proof of concept all the way to getting scaling, full-scale demonstration that these products are relevant to our environment moving forward. Now, I think I've mentioned this about some of the areas we need some products coming out, life sciences, diagnostic therapies, medical products. Clean energy, Kenya is doing fairly good in terms of a focus on clean energy, whether it is geothermal or wind That's a good thing, but we need to see all of that. Resilient crop varieties. I love my sweet potatoes like everybody else here, but we have to think about these indigenous crops. How do we make sure that we protect them? And there are even genomics ways that we can do that, improving these varieties and making sure that they don't become orphaned moving forward. For policymakers, science, they need to appreciate science is actually something that can drive economic development. Now, there are billions available every year that goes into clinical trials, but only 3 to 5% of that comes to Africa. And the best recipient is South Africa. 60% of that clinical trials actually goes to one country. Now, again, just looking at the data, the life sciences sector contributes more than almost $60 billion to the UK on an annual basis. That's a lot of money that comes from clinical trials and clinical work. If governments, or South Korea, I was there 3 months ago and you just get impressed about just how much they are doing in terms of a focus on, for example, the pharmaceutical industry there is very, very strong. That pharmaceutical sector contributing 33 billion to the economy. That's huge. That's almost like the budget of Kenya on an annual basis from one sector of science and innovation. Now, we have to make this case and make it very compelling to our governments that actually, yes, extraction way of mining is great. We can mine our gold. We don't have much of that. Whatever it is that we have by way of mines is in Kenya. But I think science can really be transformative to the economies. So just to get to my close, what have we learned from all of you as scientists across the continent? Now, we have learned that there's leadership and reforms that are required in grant making. And I alluded to a bit of that about the fact that we are trying to do unique, relevant with how we approach this. I have had many in meetings where the scientists will say, can you encourage more ambitious creative research? And I think the answer is yes, we should do that. Can we no strings attached grant? That's difficult. I don't know what no strings. I don't think there is nothing that comes to life. We not have a little bit of no strings attached, because I think there is an aspect around accountability and assurance. So you can't just get a grant and nobody then is asking you what it is that you're doing with the money. But I've heard that from the meetings that I go to. Invest in more institutions, yes, we are trying to do that. Financial support for labs, infrastructure, yes. Building science leaders, moving away from short-term projects. That's a passion for me. How much impact can you make if you're receiving a grant that is only going to be with you for 2 years? That's something we want to change that model. For example, for the large consortia that we are providing, we're giving 7 years. Because 7 years, you can start seeing some impact. And I think it is pointless to talk about impact. What impact do you get from 2 years of investment? You can only do that as a pilot. You can only do that as a proof of concept. But if you want to get into that area that I'm saying is missing in the middle of transitioning innovations, you have to think long term. And that's something I really I believe in. Incentivize in sort of more riskier, long-term science. Yeah, risky science is good, but I don't think we are quite there. Because we need to deal with some of the challenges we endure every day. But very risky science, or even blue sky sort of science, that remains a challenge. I've heard scientists say that, yes, a focus on diversifying from government, philanthropy, the private sector. Private sector sometimes is missing in action in some of the things we do. The biopharmaceutical companies, unfortunately, they see Africa still as a place to sell their products, not a place to support our R&D work in Africa, which is something that needs to change moving forward. And then, of course, one of the things I tell scientists, you don't do a good job of making a strong value proposition. I'm not saying you build unions. and you go to the streets and mapambano, mapambano. No, not that kind of thing. But I think it's good for scientists to do a very strong value proposition as to why the science of the projects is very, very important. And then we hear all the time about investing in the wide ecosystem. So what has worked for us is peer learning and co-creation. Co-creation is where we started by saying, define our priorities. We build systems, we look at building leaders, careers, and the connection between science and jobs. And not just jobs. So science actually gives dignified jobs, high-value jobs. And so, again, we have to make that case to our policymakers that if we are talking about jobs in every country, science could be a big contribution to that. Of course, science collaborations, particularly for us as a Pan-African organization, building intra-African partnerships and collaborations has been a focus area for us because, again, we see a whole scattered approach. Yet, if you want to get to impact, it is easier when you have collaborations. So the way we ask applicants is to tell us what of those partnerships or universities in different countries, what is their work stream? How does those work streams converge in terms of moving forward? And of course, value for money. Every dollar available must be accountable for. And value for money means what does it also deliver that investment? If you put money into malaria research, you put $1, you probably, the value return on investment could be $20 or $30. And that's the kind of interpretation that economics love, that kind of language. Research environments, yes, we have trying to do that, innovative blended financing. Blended financing is about going beyond philanthropy. So we need pooled funding, we need equity, we need multilateral organizations to come together to build this. The private sector and industry needs to come on board. That has never been seen. We think we are building trust, or we have built a lot of trust with the organizations and in government agencies and government departments where we have been trying to do this. And then, of course, for us, diversity, equity, and inclusivity. That will never change for us, despite the geopolitics or the politics. Equity has been in some contents, in some places, in some countries, without calling them out. Equity has been, what's the right way? It's been seen as something that actually should not be part of the conversations. But for us, this is Africa. We embrace equity, and we will continue to put that at the center. Our values as an organization, DEI, is actually embedded in that, and we're not going to change that simply because there are geopolitical shifts. So let me conclude. Good science, we say, comes from anywhere. So as an organization, we have a moral imperative to try to fill the structural gaps to support it. That is our mission, and I quote that because that was a paper written by our chief finance officer, because she's the one, of course, who handles all the money issues. And this is what we know, want to see at the end of it. It could be long-term vision, but this is what we are all looking collectively. We are looking to see a critical mass of scientists. It's about people. We are looking for places that conducive environments for doing research, policies, best practices across the science ecosystem, products, conversion of those science findings into innovations, and partnerships. Partnerships is pursuing equitable partnerships across the Africa and across the world. That's what we will then say we have strategic impact if we get there. And of course, we will need to engage across the entire ecosystem, scientists, funders, government, policy, peer, institutions, innovators, all of them. And finally, what next? Of course, we have people who are always asking, what is the next thing that is going to come out of our organization? What requests for proposals are going to come from? So we have some things that are in the pipeline. I mentioned the African Population Consortia around cohorts. The cohorts are going to have research questions. Those research questions will require funding. That is where then requests for proposals will go out at some point. Pathogen sequencing, we have an opportunity to give more grants in that area. Climate and health. The G20 coming up in South Africa, we will probably be announcing an opportunity for climate and health. Those of you who are familiar with one of our flagship programs is called Deltas Africa, developing excellence in and learning, training, Deltas, excellence in leadership training and sciences. So Deltas has been now 10 years of Deltas Africa. Is there a Deltas 3? I would say very early stages of those discussions. But let's see where we are by middle of next year. Genomics, something initiative we are calling Gen Impact. There will be opportunities there. We want to put a grand challenges call for proposals to address some of the areas all of you experience, the challenges of procurement for your supplies, for your lab. Some of it is political angle, some of it have to do with actually dealing with how we deal with bringing procurement and what does it take to do that. That will be an opportunity. And then, of course, science diplomacy. We are very passionate about how we will use science diplomacy. It was interesting to see that the State Department here in Kenya for Science is actually now embedded under the Ministry of Foreign Affairs. That's interesting, and I actually like that angle because science diplomacy can be driven by general diplomacy, and I think that's an opportunity here in Kenya, but also across the continent. We want to do more thought leadership, more science diplomacy along all the science research and innovation we are supporting. I usually don't speak in the morning, so I'm already hoarse. So thank you very much for the opportunity, and thank you for the organizers for the invitation.
Thank you very much, Thomas. Let's put our hands together and appreciate Tom. We are constrained when it comes to the issue of time, so I want to suggest that if there are issues that you'd want clarified by Professor Thomas Kariuki, you can take it up with him. Just a quick one, Tom. I know you mentioned something about mobilizing resources from the African government and also the private sector. Maybe that's something that you may want to think about developing programs for that so that you don't have a challenge in terms of sustainability. I would now like to call upon Professor Collins Ouma to now invite the next speaker, Prof.
Thank you. It's my pleasure to invite Professor Kim Yang as the next speaker. Dr. Kim Yang is a professor of biomedical engineering, University Faculty Scholar, and Showalter Faculty Scholar in Purdue University. His research focuses on data-centric biophotonics and the integration of physical and digital research technologies. He has led a uniquely broad range of projects, including cancer research, machine learning, optical imaging, spectroscopy, biomaterials, metamaterials, and cryptographic systems. Currently, he is advancing reciprocal innovation aiming to adapt mHealth technologies developed for resource-limited settings for use in developed countries, a concept recognized with the first prize in the NIH Technology Accelerator Challenge in 2020. Additionally, he has been a pioneer in cyber-physical biomedical security technologies for safeguarding pharmaceuticals with support from the AFOSR cybersecurity programs. Dr. Kim earned his PhD and MSI, which is a Master of Science in Clinical Investigation from Northwestern University, and completed postdoc training under the NCI Cancer Research Careers Program for additional so we provide site for additional information if you want to read more about Professor Kim. Let's all join hands in welcoming Professor Kim Yang. Give this presentation. Thank you.
Good morning. Let's see. Okay, my name is Young Kim from Purdue University. So the title of the talk this morning, Spectral Vision for Mobile Health Applications. I'm going to discuss a little bit about co-designing hardware and software for parsimonious AI, so you'll get to have some of idea why we are heading to this direction. I know usually I'll be mindful about the time I have, and I'll be running out of time. So I want to thank my lab members. Actually, many of the lab members are attending this conference now. Jong-woo is here. Priya actually presented her work yesterday. Sean, Atin presented his work. David. So actually feel free to talk to those members actually attending conference. They'll give you a more detailed information about our study. And obviously, I have to thank my collaborators. In particular, I want to thank my collaborator at Moi University in Eldoret, Kenya, Dr. Edwin Worry. We are doing multiple studies, and actually recently we launched a new study, preeclampsia early detection, supported by Gates Foundation. So we are thrilled to continue the study. And we have other collaboration in University of Rwanda, RBC, Rwanda Biomedical Center for malaria studies. So I will cover a few examples. So I want to just give you some background about my talk. So you heard about this. One picture is worth 1,000 words, right? Actually, I don't think this is the quotation from Albert Einstein. But if you search this, actually, you'll find this is internet. So I just included it. That doesn't mean much. But I want to change that actually photo is worth 1,000 words. Actually, as you know, if you have photo, it's just more than a picture. Has a lot of information, like color information, spatial information, and texture information. So we are really dedicated to extract this kind of information from a photo. A second thing is this, the data is the new oil. So data in the 21st century is more like oil in old days. So if you have good amount of data, you can do a good job for AI and machine learning. So we are really focused on acquiring high quality and standardized data. As you know, garbage in, garbage out. So we really do our best to quantify the standardized data acquisition and etc. And third one is actually if you look at, there's huge amount of advances in machine learning and AI, in medicine, in biomedical, in particular biomedical imaging. But if you look at actually some of our advances, actually focuses on spatial analysis. The spatial information is important, but we really want to use color information. So color information is often overlooked. So this one is just an example. Actually, color has been with us for a long time. This one is a Chevrolet chromatic circle, published in 1961, but still in biomedical imaging domain or medicine domain. We don't take advantage of color. So we really want to focus on color information for diagnostics and medical applications. So the last one is this. Actually, we are really interested in a physical AI and hybrid AI to have parsimonious. That means actually eventually we want to have edge and on-device AI. So let's look at some of the examples. If you look at a large language model, or nowadays you have a large vision model, you need billions of images, petabytes of data storage because the data size is humongous. In terms of computation, you need to have 100,000 of GPU hours. And actually, in terms of just the electricity cost, actually, it's based on the US cost. Let's say if you need train model, let's say you need a multiple gigawatt hours per model, but 1 gigawatt hour costs about $150,000, the US dollars. So that means it's very expensive. So the direction that we're really interested is we want to learn based on co-design and co-creation of software and hardware such that we don't need a big data. Algorithm and model is very straightforward to understand, so it's explainable. And also GPU and computational hours are minimal. And the impact is that speed and affordability. And also if you have actually edge computing, the privacy is another advantage we can get. So if you see actually early days of computing, old times we have a big machines like from IBM, etc. But as you all know, the Steve Jobs actually introduced personal computers. So I think this is a time from like a mega or the tech giant-driven AI to more personal AI. So that's the direction I'm very interested in. And half joking, my biggest competitor is Google. And this is some of the research Google is doing. They're very interested in retinal imaging. Retina is the back of the eyes, has a lot of information for diagnostics, many other applications. And also they're very interested in take the photo of eyes, external eyes. So this is some figures from Google website. This is what they do. Take photo, just one example, and they have humongous network. And even they trying to learn from EHR. And the EHR, we have a clinical laboratory result. So they want to see actually they can even learn clinical laboratory result. Actually, they're doing pretty well. But one of the limitations they have is purely data-driven. So this is a paper Google published like 2 years ago. But if you look at the number of the data, actually the number of patients they actually require to train such a big model is 300,000 patients. And as a laboratory scale and a university scale, actually I cannot afford such a massive amount of patient data. So then what should we do? And as an engineer, actually we know physics, we know biology, right? So the idea is let's kind of incorporate our domain knowledge into a learning algorithm so that we don't need a big data. Actually, everything is very manageable. In our lab, our domain knowledge is tissue optics, light tissue interaction, and also we are developing physical element, and there's a color chart, color reference chart, and etc. So by co-designing physical elements and algorithm or model together, and actually we can have a very affordable AI. And one of the beauties that, again, we don't need big training data sets required to develop AI. So we are really interested in moving on to edge or on-device AI compared to other kind of conventional approaches driven by tech giant. So we have some color charts. Actually, we saw some of cases from the talk yesterday. This is specially designed. It's co-designed actually for AI algorithm. So what we do is hyperspectral imaging. The concept is very simple. So you want to take images for different colors using different filters. Basically, we have a three-dimensional data set. It is called hyperspectral cube. So if you look at each pixel, we have a spectral information, so we can get additional functional or physiological informations. If you look at our photo, because we have three color information, green, red, green, and blue, in a sense, it's a multispectral imaging. But as you know, the spectral information is very limited. So the simplest way to get hyperspectral imaging data is just use some kind of filter and take multiple images. But as you can guess, it's very slow and it's bulky. So a long time back, even we built our own hyperspectral imaging systems, but it's big because we need to have multiple optical components. The beauty is, again, we can actually get spectral information. So nowadays, we really want to combine hyperspectral imaging with mobile health. So do you think it makes sense to attach this bulky system to a smartphone? It's a no-brainer. Definitely no. So we are struggling to doing the hyperspectral imaging system without using hardware. So this is kind of forward direction. So this is hyperspectral imaging data, or it's actually we have in nature. And if you have a smartphone, actually smartphone has like spectral response functions or the spectral specificity functions. So actually convert to 3 color information, RGB. So this is the forward calculation. And depends on your camera model, you can have a slightly different color because the sensor has different spectral response functions. So this is forward direction. Then the idea is it possible to go backward direction? So this is, as you can see, ill-posed problem because we have three kinds of information, but what we want to get is all detailed color information. But mathematically, there are many different approaches like L1, L2, and even we can use deep learning. So we are really interested in doing, developing algorithms to actually solve this ill-posed problem. That means actually we don't need a big system. So another challenge we have is actually when you use a photo, if you take photo under different light conditions, and if you use different devices, and even different file formats to save, color changes. So we have to overcome this limitation. We have to get absolute color. So we developed the color chart. It's actually specific developed for a specific application. Include this color chart when you take a photo. So basic idea is if our model or algorithm learns the color of color chart, and actually we can correct the color for the area of interest. So we name this as one-shot learning because we can do a similar thing using data-driven approach. For example, let's say you have thousands of images. You can develop a model to correct color, but again, this is a purely data-driven research, and here we don't use any trained model. We train the model per single shot. So because we just need one image, because we have this color chart, physical element, so our algorithm is very manageable, and now we want to implement this into edge computing, on-device computation. Again, we don't have to use trained algorithm. We want to train algorithm per image. So this is one other example. So we have a color chart here. If the algorithm learns about these colors related to inflammation, and actually we can map out inflammation directly. So this is one example for cancer research. And also we are developing different types of color chart with wide color gamut, so that if we include it in the photo, we can get hyperspectral information, basically different wavelengths information, without using hyperspectral imaging system. So this is one example, co-design or co-creation of physical elements and the model or algorithm, and we can actually achieve highly manageable AI, which is quite different from a conventional AI you've seen nowadays. So this one shows some other examples. So this is a chick embryo sample. So if you have a, let's say the algorithm is trained and it can extract oxygenated, deoxygenated, and if you have both, actually you can get oxygenation saturation. But again, only data we need one photo, like one RGB image. So this is a validation using conventional hyperspectral imaging system. And another thing we are really thrilled is that actually conjunctiva. And we all know that retina, the back of the eye, has a lot of information. Like you can do full range of diagnostics if you have a fundus image. But we actually now see that the conjunctiva has very similar information, like a mirroring retina. The biggest advantage of the conjunctiva image is that we don't need specialized equipment, only because there are a lot of microvasculatures, so we are really analyzing this using the AI method we are working on. So one example, so we just can take a video of the inner eyelid, and this one actually we want to correlate it with the functional near-infrared spectroscopy, kind of see some similarities between the brain function. So if you look at the phase difference between oxy and deoxy, actually it really matches even with the brain activities. And another active area is to measuring blood hemoglobin in a non-invasive manner. The best way is to get a blood draw or finger prick technology. And compared to oximetry, actually measuring absolute blood content, it's a challenge. And as you can see, it's a lot of applications. So take the photo of the inner eyelid, and we do spectral reconstruction. So from RGB, you have a hyperspectral. And if you have a spectrum, because blood hemoglobin has a very unique spectral features, we can compute blood hemoglobin. So we have this algorithm, and luckily, due to the collaboration with Global Health Labs, actually it's a part of Gates Venture, so we really want to translate this into edge computing, because in the US or some Western settings, we have good cellular connectivity, so we can rely on cloud computing, but oftentimes we don't want to use actually cloud computing. So we want to do this on device. So this is the direction we are working on. And this is one example from the US study. So we ask a patient, sickle cell patient, take photo every day, and we can compute blood hemoglobin level. And actually, if you look at, I'm sorry. Oh, how can I go back? I'm sorry. And actually, we can see some changes during crisis period. So this is one good example that can be used in digital health, even in the US. And another study we are doing is malaria detection, but we want to detect malaria in a non-invasive manner. As you all know better, malaria is a microvasculature disease. So this is the non-malaria case that the eyelid from school-age children in Rwanda. And if you look at malaria cases, it's really difficult to kind of pinpoint out the differences, but there are some differences. So this is a really good problem to solve using machine learning and AI. So we apply this to radiomics and apply some advanced machine learning classification algorithm. So this is some of our information about the feature extraction and etc. from radiomics. And this is overall performance. Again, based on the photo of the eyelid, our detection performance, actually our gold standard here is the rapid diagnostics. And we can achieve area under curve from ROC curve is about 76 up to like 68% to 84%. So this can be used as a pre-screening tool or just identify someone who will benefit from actual fingerprint. So we're thrilled about this result. So this is actually a study with the University of Rwanda in two different districts. So I'll actually explain a little bit more. Another interesting area is that actually we can detect hypertension as well. There's a very unique differences. So we applied machine learning. So this one, you can actually detect the stage 1 hypertension with 85% area under curve. And if you add a BMI, so this one actually we add a BMI as an independent predictor, as you know, it's a risk factor, then actually the performance increases. And you got actually Priya presented this yesterday. We also look at the bulbar conjunctiva, and we can visualize individual microvessels. So as you can see, there's some changes in microvasculatures in bulbar conjunctiva, and statistically, we saw differences, vessel diameter and tortuosity. And again, only thing we need is smartphone images. And this one is the prediction of hypertension, including BMI. So we have two active global health research studies. One is, I mentioned, the malaria detection. I don't need to explain why I need to study malaria. So this is our ongoing study in Rwanda with collaboration with the Ministry of Health, RBC, and the Ministry of Education. One of the things in the study is that actually we want to train school teachers to be community health workers. So that's why we need to get a support from the Ministry of Education, because we are now working with school teachers. So we launched two studies in two different districts, Bugesera and Gisagara districts, and so we trained school teachers, so they serve as community health workers. They know how to do finger prick for RDT, HemoQ, the blood hemoglobin test, and also they are collecting dry blood samples and smears, etc. And in addition to those, actually they're trained to get high quality photos of the eyes, so we can actually refine our algorithm. Some of photos that we went to different high schools near the health centers or district hospitals and trained the school teachers. Again, we need to get high quality photos, so this training is essential. And another active study is in Kenya, working with Moi University, Moi Teaching and Referral Hospital in Eldoret. Here the focus is maternal health. Actually, we are developing blood hemoglobin detection for pregnant women. And another thing we are really thrilled is that actually, as you know, preeclampsia, the hypertension is a very important component, and we saw actually the microvascular changes associated with some hypertensive disorder. So we want to actually develop a prediction model, like early detection or stratification of preeclampsia at Moi University, working with Dr. Edwin. So we are really thrilled. Actually, recently we got support from Gates Foundation to launch a relatively big clinical study. So I'll be wrapping up my talk. From my perspective or US perspective, maybe think that this is one way. But I really want to point out that actually if you look at rural areas in the US, we don't have hospitals, we don't have a really good healthcare infrastructure either. So this one is the idea, like a reciprocal innovation. So it's develop technologies here, and my job is that I need to bring these technologies back to US because we have a really good momentum for digital health and mobile health. Actually, whenever I apply NIH, US NIH grant applications, they really want to make sure that actually I bring the technologies developed here bring back to US because we need in the US these kind of technologies as well. So the idea is a reciprocal innovation. So I think I want you to think that actually it's a win-win situation. So let me conclude my talk. So we are really interested in hybrid machine learning, so combine data-driven approach and as well as some kind of modeling, like including domain knowledge into algorithm. So the advantage is definitely we don't need a big data, but from the AI standpoint, it's transparency, and also we can understand a little bit of our model. So that's a huge advantage for adaptation. This one is actually, again, hardware and software co-design or co-creation. The idea is that, again, we want to move toward edge or on-device AI by optimizing our parsimonious computation algorithms and etc. So as you can see, there are multiple advantages. Privacy, because actually everything runs on a device, privacy, and also affordability and speed. So actually we can get several different advantages going in this direction. And finally, as an engineer, I'm very interested in building things, but for a rapid adaptation and translation, I realized that actually I need to minimize hardware complexity. But as you know, nowadays, everyone has smartphones, and smartphones have very high-end imaging sensors and etc. So the idea is let's minimize hardware complexity and just take advantage of what we have for medical diagnostics. So thank you, and I'm happy to take any comments and questions. And once again, I want to thank you, the conference organizers, for hospitality, and in particular, Dr. Lucy Ochola and Dr. Kennedy Okio for a kind invitation. Thank you.
I think that we'll because we are trying to manage time, if you have any comments or questions, we'll keep them for later. Professor Kim is with us here for the whole of today, and I hope that he should be able to address some of those questions or concerns. Thank you. I want to invite Dr. Daniel Chai to introduce the next speaker. Thank you.
morning. I'm from the Kenya Institute of Plant Research. I'm a veterinarian, and I'd like to welcome you to the 6th plenary talk, which will be done by Dr. Javier Gulen. And may I quickly introduce him. Dr. Javier Gulen is a veterinarian and is the ALAC International Senior Director for Europe, Latin America, and is responsible for Africa and the Middle East. He is a member of the board of the Laboratory Animal Limited, which publishes the journal Laboratory Animals, and of EAR, European Animal Research Association, and is the former president of FELASA, the Federation of European Laboratory Animal Sciences Associations, and SECAL, the Spanish Association for Laboratory Animal Science, and was also governing board member of ICLAS, International Council for Laboratory Animal Science. He has published numerous articles and chapters on legislation, ethics, and quality, including 3 books on regulations, recommendations, and management of research animal care and use program. Welcome, Dr. Gulen.
Thank you very much, Daniel. I would like first to thank HIPRE for the invitation to come here, and especially Drs. Peter Huetera, Lucy Ochola, and Daniel Chai, and also Ms. Lucelaine Bogori, because of her logistical, let's say, help. It's a pleasure to be here, and it is my first time in Kenya. A wonderful experience. Congratulations for this very nice event. So let's go with the presentation. No, that's not the first one. Here we go. Okay, we are going now to talk about quality, animal welfare, and ethics in science, especially on animal research, and I will try to focus a little bit at some point in non-human primate animal care and use programs. So why quality in animal research? We will be discussing a little bit about why we may be interested, you may be interested in implementing quality in animal research, how this is linked, quality, to animal welfare as well, and I will explain a little bit our accreditation system called ALAC International, and I will be mixing a little bit some of these concepts during my presentation. So why? Why can we, in general, in the scientific community, interested in applying or implementing quality in our animal research? From the moment you researchers have an idea, you use your animals to produce drugs, to develop drugs, to publish in a recognized journal, even to perform surgical training for medical doctors, for example, or to produce, to develop medical devices. From the scientific point of view, there are many factors that may influence the outcome of your research. Maybe the health status of your animals, genetics, the environment, the experimental design and procedures. That's for me a very, 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 very important point, probably the most important, I would say. Husbandry, not only the experimental procedures, but also the husbandry, the procedures involved in the animal care on a daily basis, and very importantly, the competence of the staff, both performing the experiments and taking care of those animals. We are in a, let's say, critical moment in animal research because of the pressure, societal concerns, as we will see in a moment, and we are being criticized, or the scientific community is being criticized in part because of the sometimes poor reproducibility, sometimes poor translatability of what we are doing with the animals to produce these things, right? So this combination of quality, animal welfare, and ethics is extremely important for the animal research community. Because as I'm telling you, there is a lot of pressure, and that societal pressure goes into legislation in many countries. So we may want to implement quality also to comply with legal requirements, more and more strict every year in many countries, especially in Western Europe, for example, and in other countries as well. So this, again, this combination of scientific and ethical interest is very clear. When we talk about those potential interferences in our research, scientific interferences, they are also linked to the ethical use of the animals. Because if we are not producing quality science, we may be wasting public money, for example, and in addition to that, when we are using animals in research, we may be we may be producing certain unnecessary harm to animals, which is also a very important ethical concern. And society is more and more worried, concerned about the quality or how, why, in which conditions, and for what purposes we may be using animals in research. And of course, there are institutions that may want an international recognition. In some cases, it may be a commercial interest, and then some institutions may be willing or interested in applying ISO, for example, in any kind of environment. If in some institutions want to perform certain type of experiments in the preclinical development of drugs, typically toxicology, safety studies, they must implement GLP, good laboratory practices. And I will explain the ALAC accreditation process in a minute, which is the one that it is mainly focused on animal care and use programs. So in the end, I will explain all these different customers that we may want to satisfy when doing animal research. And I will explain it from the ALAC point of view, having the animal as our first customer. If we want to do things well with our animal, we may be satisfying the rest of the potential customers that we have when we do animal research. For example, the legislation, as I said previously. So if we focus on doing things well with the animals, our competent authorities, regulatory bodies will be happy with our institution. We may be satisfying the buyer of a product or service. For example, quality is very widely applied in breeders, animal breeders, because they are selling a product, they are selling animals, or contract research organizations may be interested in selling their studies, their services to other companies. Also, it may be interesting to satisfy funding sources, especially in the US. For example, ALAC accreditation is quite, let's say, interesting to facilitate certain type of funding. But if we are implementing quality, the funding sources will trust us better, and they may be more interested, especially sometimes in this globalized world, right? In where collaborations are spreading out a lot. And then we may be satisfying society if we do things well, and in these times when societal concerns are increasing, we can tell society, look, we do things well because we have implemented this certification or accreditation scheme. And in the end, if we satisfy all these other customers, we will be satisfying you, the research community. If we create in our institution a better quality environment, researchers may benefit greatly from all this environment, to publish, to get funding, to do things well, to produce better quality science, right? So everything is linked, scientific interest and ethics and quality. And this, let's say, trend to improve the quality, I remember the first speaker this morning was mentioning the excellence in research. That's a very important point. It is very linked to some ethical principles. I think we are all well aware of the basic ethical framework where animal research lives in, which is based on the principles of the 3 R's, replacement, reduction, and refinement, that were more or less described more than 60 years ago by these two people, Russell and Barch. And there are also other type of ethical principles that can also and are also applied in our environment, even if they were, let's say, born in the agricultural environment. In the UK, the 5 freedoms, these ones that you can see there, that have evolved now in what we call the 5 domains that pay more attention to the mental state, the behavioral interactions of the animal. So there is an ethical framework that it is going hand by hand with the improvement of the quality of science. And I always like to highlight this, and I was discussing with this yesterday at lunch. This article published almost 30 years ago, where the title is very interesting. Happy animals make good science. And in this article was one of the, in those times, that was trying to promote the ideas that I am explaining here today based on a better experimental design using right species, appropriate numbers, sexes, again, paying attention to the reproducibility and translatability of the research that we are performing, use of healthy animals, appropriate environment, not only experimental procedures, but husbandry procedures as well, and very importantly, the competence of personnel. All these aspects are very important to link the welfare of the animals with the quality of our science. And what quality systems we may have? The first quality system is yourselves, is your institution. It is the standards that you want to implement in your own institution by your quality assurance unit, by your IACUC, Institutional Animal Care and Use Committee, or similar, by your management, by your veterinary team. You are the first ones interested in applying quality in your environment. But if you want to go to external providers of quality, you have the systems, ISO can be implemented anywhere, all kind of things, including animal research. It is not specialized. GLP, good laboratory practice, for certain type of studies is a mandatory thing. The big difference between those and ALAC International is that ISO and GLP are not specialized in animal research, so you are not going to learn much on how to improve animal welfare in your programs from them. So basically, ISO and GLP, if you have experience and they visit you, they will check in your institutions that you do what you say you do, but they may be not challenging if what you do is good for your animals in certain ways. I will explain ALAC International, which is not by definition a quality system, but it shares a number of features. And there are others like GMP necessary in other type of development of materials, or equipped is another system that is being developed in these days. So I will explain now, as I told you, what ALAC International is about, especially because they pay my salary, okay? And that's why I'm here. And you can see there in blue three important characteristics. We are a private organization, we are a non-profit, but we are a fee-for-service non-profit. We charge for the service, okay? And it is a voluntary accreditation program. It is a voluntary thing. We don't represent any government or agency. We are managed by professionals, and as I will explain in a second. And nowadays we have more than 1,150 accredited programs in 52 countries, geographical areas all around the place. Some of them, not many, just a few, in Africa, especially in Mauritius, related with non-human primate breeders. How is our structure? And this is a very important point, because we are managed by the scientific community. We have a number of scientific member organizations that you can see there in green. From there is, as in all organizations, a board of directors that tell us, the staff, tell us the strategy to follow, and we do the operations. We have the headquarters in the US. From Europe, I run Europe, Latin America, Africa, and the Middle East, and there is another one in Asia Pacific, in Thailand. And the people doing the evaluations on the ground are independent professionals with high skills, very good recognized professionals. Here we have one, for example, Dr. Yang Hao, who collaborate with us on a voluntary basis to go to the institutions and evaluate. They are highly recognized professionals in the field. And they are helped by ad hoc consultants, who are the same type of professionals. They may be working in academia, in pharma companies, in CROs, in institutes of research, anywhere. So these are the people, real professional. That's the big difference with other systems. These are specialized people. So, and the very important thing is that we accredit not a building, we accredit an animal care and use program with all the elements. This is the definition in the Guide for the Care and Use of Laboratory Animals, which is one of our primary standards. And this applies, as you can see in blue, to all the elements of an animal care and use program. Remember, we don't create a physical structure. We create an animal care and use program. And an animal care and use program includes individual responsibilities within the institution, the competence of the staff, not only using but also taking care of the animals, the oversight and ethical review process. In many countries, this is performed by the institutional animal care and use committees. There can be ethics committees, animal welfare bodies. They may be called in a different manner. The health and safety of personnel, that's also an important point of the program. Everything relating to the daily care of the animals, how they are housed, fed, sanitized, etc., the enclosures, the program of veterinary care, one of the most important points, and the facility. As you can see, the facility, the physical plant, is only one part of the things that we will evaluate and accredit as part of the program. This has to be, in practical terms, described by the institution in what we call the program description that follows the outline, more or less, of this primary standard. And there you will see like 4 main areas that an institution will have to describe in a very long questionnaire to us, and that we will read and evaluate on site when we do a site visit. So we will go on site after reviewing the program description of that institution, where they have to describe and explain how they do all these things. Then the institution will complete the program description. We have a particular European format in case for a European institution. This is a very interesting self-evaluation tool. This program description template is freely available in our website. You are free to download it, and it is a wonderful self-evaluation tool just for you to review all the items that you can think about. Then we assign the site visit to a council member, and the council member with X numbers of ad hoc specialists or consultants, the same type of professionals, will go into a place and see on site how things are being performed. So in the end, they will produce a report, an evaluation report, that after some electronic reviews by other council members, it will be discussed at the face-to-face Council on Accreditation meeting. It is a peer review process by professionals, and they will decide what to do, and they will communicate it to the institution. This process is repeated every 3 years, so it's a continuous improvement process, right? There are other requirements to produce an annual report, to report to us adverse events if happens, but the important thing is that the process is that specialized people will go to your place and evaluate on site how you do your animal research. And for that, we try to ensure that institutions comply with applicable legislation at a minimum, but also we have 3 primary standards and other documents that we use as references to benchmark the activities of the institution. And as I will explain in a moment, what we try is to, in addition to ensure that institutions comply with the engineering standards that are described normally in legislation, they comply with the performance standards. I'm not sure if you are familiar with this concept. I will try to explain it in a minute. Engineering standards are those standards that are fixed, that one can measure. Okay? Frequently, as legal requirements, the most typical one is the minimum cage space or the minimum space that animals must have. That's measurable. A cage for any human primate, a mouse, or whatever. That's an engineering standard that we have to comply with. But we also like to apply what we call performance standards, which are based on the outcome of the process. We admit that and acknowledge that there may be many different ways to achieve a good outcome. And that's what we try also to evaluate, applying professional judgment, as you are seeing there, and trying to evaluate the outcome of our process. I will show this in a very simple example. What would you think it is best to clean cages? These are just rodent cages. When I ask this question, normally we don't have time here to discuss today, but many people say it is much better to use an automated machine, right? And I say, not necessarily, that depends, because you may be using a sophisticated automated machine, but if you are not using it well, and you are not evaluating that the cages are clean periodically, you may not be cleaning your cages well. On the other hand, you may be manually cleaning cages, but if you do it well and you periodically evaluate that your cages are clean, you are doing well. So there may be many different ways to do things. This is a very simple example. It can be applicable to many more complicated things. The important thing is that you demonstrate that the product, the outcome, is good. If you want to see about our standards in the AILAC website, we have the 3 primary standards that I've commented. We have position statements that give real strong, let's say, standards, in our opinion. In addition to those, we have other guidance statements. They are freely available in our website. It can be useful for you in some occasions. We have a number of other documents called reference resources that we can use to help us during the evaluation as well, and some frequently asked questions that may be useful for anyone interested. For non-human primates, in practical terms, in these documents that I'm referring to right now, there is a position statement. Position statements are strong messages to the community, and for example, we have one on B virus, and here you can see a must, which is a mandatory thing for us, very important. So, for example, when we visit institutions using breeding macaques, for example, we expect them to have a program to control, to monitor the B virus because of as part of the health and safety program. And we have now a guidance statement on legal acquisition and use of wild-caught animals. I know this is a very controversial issue. We had a good conversation yesterday over lunch. And what we expect, and you can see there, is that there must be a robust justification when that happens. This is a very controversial issue. For example, ethically, in the European Union, for example, it is forbidden. We have to use F2 animals when referring to non-human primates. I know that in many other countries, this may not be possible, and there are other ways. And so we evaluate that very carefully on a case-by-case basis because of the ethical and scientific reasons. So basically, just to finish, AILAC represents quality. It's a self-evaluation process. You may want to challenge tradition when you self-evaluate yourself. You would get engaged in a continuous improvement process. The evaluation is absolutely confidential. Peer review is external. And I like something which is interesting. It promotes team building. You will have to work with other people in your institution, health and safety, maintenance, the animal service, obviously. If you have a quality assurance unit, is a group who may be interested, your IACUC or ethics committee or whatever you want to call it, management. If management is not convinced, no way to make it. You need the support of management, no doubt. And there is other let's say, benefits. You will be improving animal welfare, the promotion of the 3 Rs. I like the animal care people, the people in your institutions, if you do research, taking care of the animals, do a very hard job. And when they feel that they are contributing to an international recognition, it is a very good thing for them as well. We help promoting scientific validity, the robustness of the research produced with our animals. We demonstrate accountability to the society. Remember, in many countries, performing animal research is becoming more and more difficult, and that will happen anywhere. It may facilitate funding, especially in the US, and it provides assurance in a global marketplace. Nowadays, there are more and more international collaborations. Pharmaceutical companies, for example, are diminishing the sizes of their structures and subcontracting almost everything. So, and they rely on this type of trust guarantee that they may get from an international organization. And what we do, I was telling you that the other quality system will check that you do what you say you do. The difference with AILAC is that we check that if what you do is good for animals and science. That is a little bit the concept. If we could represent this in a graphic, quality level in different countries because of different legal requirements sometimes differ significantly with an international accreditation. I will never put this at the same level because we are humans, there are different environments, but we help harmonizing not only animal welfare, but the quality of science. And in global terms, thinking internationally, we think that this is a very important point. And yes, Kenya is in the picture too. I remember, for example, Daniel participating. I invited Daniel from Kipra in 2022 in the Federation of European Laboratory Animal Science Association Congress to explain how to develop a good animal care and use program in the African environment. So my final slide, when we work together, researchers, animal care personnel, including technicians, veterinarians, everybody together with our research animals, we will be helping to produce good science. And that is basically the main concept. Thank you very much. Don't know if there is time for questions or not.
Thank you, Dr. Gulen. Any question? I know we've run out of time. We should be in the middle of the break. Any burning question, one or two? Thank you. Now it's my pleasure to, thank you. Yeah, let's give him a better clap than that. I've known Dr. Gulen since 2011, online. But yesterday we met physically. Okay. Now, for the break, it's almost, it's 10:20 now. Ideally, we should be coming back in 10 minutes from now, okay? Now, what I propose is that as we do the break, we try to do it maybe for 15 minutes. Okay. Before that, there is a poster announcement. Number one, all judges to hand their evaluations. 2, yesterday's presenters to pull down their posters. 3, today's presenters to put up their posters before lunch. 4, anyone who did not present yesterday to see Dr. Ruth or Rhoda for directions on where to set up their posters. So after the break, which we propose, I think if we do it for 15 minutes, let's do it 15 minutes. So we take our tea first, then by that time this hall will be partitioned. And Dr. Ochola, as we partitioned, room A will be on this side. Lucy? Room A will be on this side. Room A will be on this side. So room A will be this side and room B will be this side. So let's go for the tea break. Let's take short time possible. Thank you.
hivi. Winnie ako wapi? I have not gone to the gym that
Hello. So good morning, everyone. I hope you all are doing good and enjoying this conference. So we are on the parallel session now, which is number 8. And this session is all about flow cytometry and clinical applications. So we are going to have 7 talks in this session up to the lunch break. And today's the first speaker of this session is Dr. William Talford. Before I invite him, I would like to introduce him formally to all of you here. Dr. Talford received his PhD in microbiology from Michigan State University, USA in 1994, where his laboratory developed some of the earliest techniques for flow cytometric detection of apoptosis. He became a staff scientist at the National Cancer Institute, NCI, NIH, USA in 1999, and is currently a senior associate scientist and director of the flow cytometry core laboratory in NCI Laboratory of Pathology. Dr. Talford's main research interests include advanced instrument and assay design and development aimed at solving unusual problems in flow cytometry and cell biology. And I also like to say that he's pioneer in helping people with the flow cytometry needs. And I think today's talk that he's going to give is about building up a flow cytometry facility in one of the universities in Kenya. So thank you, Bill, for joining us today, and I'll now invite you to deliver your talk. Thank you.
Thank you so much, Hamid, for that kind invitation, and thank you to Dr. Lucy for inviting me to be here. If I can have the first slide, actually. There we go. Yeah, that's fine. Great. So just to tell you a bit about myself, I run a flow cytometry core laboratory at the National Cancer Institute. I've been there for over 20 years. And when I first arrived there, we started a program dedicated to donating used refurbished equipment abroad. I was always appalled by the amount of equipment at the NIH and other institutions that is used, is still perfectly functional, and then ends up basically in surplus once it's no longer needed. And it turned out that the NCI actually had a mechanism for donating equipment abroad. So our goal has been to place refurbished cytometers in not only resource-limited laboratories, but also laboratories that are not necessarily accustomed to doing flow cytometry, but could benefit from the technology. Now, with all the changes that obviously are occurring at the US right now, we are actually, the program is really no longer functional at the NIH. So we're transitioning it to the International Society for the Advancement of Cytometry, ISAC. And we formed something called the Instruments for Science Task Force. Hemen is a member of this. In the past, we used this donation mechanism that the NIH had. Now we're collecting instruments from industry and academic laboratories. So currently we have nearly 40 instruments that we have placed abroad. And each of these little dots down here has a story behind it. I'm going to tell you a few of those stories. This is not even quite the full list. You can see we have instruments on all 6 continents, including North America. We really started in Russia and Europe, but since then, we've moved on to the rest of the world. For those of you who are familiar with Seeding Labs out of Boston, we collaborate extensively with them. Seeding Labs prepares these instrument packages. They have an awards program where they supply laboratories with entire suites of equipment, and we are currently the source of cytometers for them. So we collaborate extensively with them. And as you can also now see, ISAC is taking more and more responsibility to place these donations. So I'm going to go way back in time. This is 2002. I had a colleague in Russia who desperately needed a flow cytometer, Alexander Sapozhnikov and Yelena Kovalenko over there in the upper left-hand corner. We gave them a Coulter XL cytometer. And it really formed the nucleus of a core laboratory. because then we were able to go on and add additional pieces of equipment to their lab. We put in a fax scan, a fax scalper, a fax Vantage cell sorter. Almost all of this equipment is still running today. We've gotten very good at maintaining the equipment ourselves, keeping it running. And this is, again, not just something that we've set up on a charity basis. We collaborate extensively with this group on a number of projects in basic immunology. Now, those were happier times. Obviously, the situation is more difficult now, but we still try to support this group virtually as best we can, and I really hope someday I can go back. I've been there many, many times. But for those of you who have been in Flow for a while, that's a Fax Vantage cell sorter still running as of 2025. This is a collaboration we're really, really pleased with. We gave a fax scalper cytometer to the Central Department of Biotechnology at Tribhuvan University in Nepal back in 2017. They use this instrument for immune monitoring. This is an infectious disease lab. They work on dengue, equine encephalitis. They did a lot of support for COVID during the pandemic. So once the instrument got there, we have organized a number, through the live education program of ISAC, we've organized a number of workshops. There have been 3 live ones so far. You can see Hement down there in the lower left photo. But it's been a really terrific collaboration between these groups, between our group and theirs. We've also placed an instrument at the Center for Health and Disease Studies in Nepal. This is also in Kathmandu. They are a nonprofit organization that provides laboratory facilities to clinical investigators who want to do research but don't have access to labs. And they've gotten a caliper as well. And we continue the follow-up. This is a collaboration that's been going on for 8 years. So I was just there back in January and April of 2005. Paul Wallace is in the green puffer coat there. He's a former ISAC president, ran the core facility at Roswell Park in New York, has really adopted this group and has helped them polish a lot of their techniques in immunology. We're also upgrading the equipment as much as we possibly can. And now we're trying to get them a newer instrument. They're really ready to move on beyond 4 colors. We have 2 instruments in Ukraine, both donated before the war. This is a caliper that was given to Taras Shevchenko University in 2019. This is a group that, they're actually chemists. They build antibiotics that can attack bacterial biofilms. So they needed an instrument where they could do testing on toxicity. So they've got a caliper there. Andrea Kosariza, who is going to be speaking after Hement, was actually the person who spearheaded. He has a collaborations with many groups in Kiev, and spearheaded this particular donation. We also have an Accuray C6 in Ternopil, which is in the western part of the country. They study Lyme disease. 60% of the logging workers in Ukraine test positive for antibodies for Lyme. It's a huge public health problem. So they wanted a way to be able to do immune surveillance on these patients. We've never been able to visit, unfortunately, between the pandemic and the current war. We've never actually gotten there, but we've done a lot of remote training through Zoom to help them get up and operating on their equipment. This is one we're particularly proud of. This is the School of Agriculture and Biotechnology at the University of Eldoret here in Kenya. This young lady in the upper right-hand photo, that's Dr. Emmy Chepkowish. She is a professor and an expert in plant genetics and molecular biology. They also won a Seeding Labs award, and even though she had no prior experience in flow, she asked for a cytometer because she wanted to be able to do plant ploidy and genome sizing. So they got their machine. Again, it arrived during the pandemic. We had to wait a few years to put it in, but we installed it in June of last year. And ISAC members have gone there to help train them on the instrument. That's standing next to Emmy, that's Sasha Makarowski. She's the former director of the Flow Core at the Curie Institute in France. So the machine went in last year, and what we've really tried to do is then send in experts in the technique that the group is interested in. And our expert here is Hement. He is extremely knowledgeable in plant analysis, ploidy, and genome sizing. So he and I went there in October of 24 to help get this group acquainted with techniques in preparing plant nuclei for DNA content analysis. And I was just there a week ago. They're pretty much doing it on their own now. This is actually cassava nuclei, both a wild type and a mutant strain, and you can see the shift in the DNA content. Hement is going to be there next week. He's going to bring them size standards, genome size standards, so they can start quantifying this and determining the actual size of the plant genomes. This technique is critical in plant breeding and selection. And this group, they do very sophisticated molecular biology and plant selection, but this technology will hopefully enhance this. This is something that breeders and this is information that breeders and producers want to know. We currently have 2 instruments in Ghana, one at the University of Cape Coast in the pharmacology department there. Robert Peter Binet is the PI. We also have an instrument in the Department of Anatomy at the University of Ghana. These were both, again, shipped during the pandemic, so we only installed them last year. The Cape Coast group is interested in doing toxicology and viability and cell stress studies. The anatomy group wants to do cell cycle on embedded tissue. And I'll actually be with both of these groups in about a week and a half to follow up with them. We have a 3rd instrument also going to the Center for Plant Medicinal Research up in Mampong. We're going to do the install next week, and we're going to do a concurrent workshop as well. Now, as I told you, the instruments historically have come from the government, are from used NIH equipment. That's really no longer possible. So we're starting to accept donations now from companies and academic institutions in the US. Insight, which is an analysis company up in Delaware in the States, recently donated an X20 to us, a BD X20. This is a very high dimensional instrument, 18 colors, a very sophisticated instrument. And it turned out we had a member of ISAC, Roxana Del Rio Guerra, who had a collaboration. She's at the University of Vermont. She had a collaboration with the Center for Infectious Disease Research in Zambia. So we arranged to have the instrument sent there and installed. And this actually kind of highlights the responsibilities on our side, because this machine has been a little troublesome. We've had trouble keeping it. It's the newest, most sophisticated machine we've donated, and it's also been the most trouble. So we've made 2 visits there so far. We're going to actually send an engineer there this winter to try to get this problem resolved. And it really illustrates that once you send these things, you're kind of responsible for them. You can't just let them sit. But this is a terrific immunology. They do very sophisticated immunology there. It's a very well-equipped institute, and they need the full capabilities of this instrument. They need to do high-dimensional flow cytometry. We have other instruments in Europe, and this is a wonderful example of how these groups leverage these donations. This is the Institute of Molecular Biology in Armenia. Gaia Manyukin is the PI, MD, PhD is the PI of the lab. We gave them a caliper in 2018 through Seeding Labs. We then gave them an LSR2 in 2021. This was a direct NIH donation. And since then, they've gone on to buy their own equipment. They've taken these instruments, they've generated data, they've published papers, and they've gone out looking for money. And they've really run with the opportunity that they've been given here. And we've done workshops with them as well. We did our first donation about a year and a half ago to South America. This is the Institute for Health Sciences in Paraguay. They needed an instrument for doing actually pediatric immunodeficiency analysis, so we gave them a caliper. They already had a Canto, but it was not in their program. They didn't have easy access to it. So now they've got their own instrument to do this. And you know, once we get these people involved, then they become we pull them into the group, and they become part of the support structure in their particular part of the world. We're also trying to get creative about how to get equipment to the place that it needs to go. We do have a shop space in Maryland where we work to refurbish, get the equipment ready to go. But for really big stuff, we're trying to restore it where it is originally located from the original donor, and then ship it directly to the country where it's intended or to seeding labs where they will take care of the importation for us. It turned out a couple of years ago that Yale University decided to retire all of their LSR2s. For those of you who are not familiar with this, this is an enormous instrument, very capable, but very, very big and very heavy. So we were able to set up a system where we actually restored the instrument at Yale and then sent them a crate, and they were able to send it on to Boston, where it would go on to its final destination. This particular instrument is actually going to the University of KwaZulu-Natal in Durban, South Africa. There's an immunology group there that currently has to drive 3 hours to get to a flow cytometer. So it'll be very nice for them to have their own instrument on site as well. The donation we're most excited about, that still hasn't happened yet, is to Bhutan. This is Bimal Chetri. He is a plant biologist at the Royal University of Bhutan. He did his PhD in India with Latha Rangan, who's a friend of both mine and Hement's. Bhutan currently has no research cytometers at all. They have a few clinical machines, but nothing research-wise. So he approached us about getting a machine. We were able to get an instrument surplus company that does these restorations to donate a BD Accuray C6, which is this instrument right here. If you're not familiar with this machine, it's a 4-color instrument, similar in capability to the Caliber, but very small, very simple to run. All he wants to do is plant DNA analysis. This is the ideal machine for him. And importing things into Bhutan is not easy. Sending big stuff is hard. So we're sending him a small instrument that really is well within what he needs to do the analysis that he's after. Bimal studies indigenous plants, does genome characterization on them. We're really hoping this will be a nice addition to his laboratory. So how do we do this? Well, we get the instruments, whether from government or industry or from academic sources. We have a crating system. We ship them, we arrive, and then we try very, very hard to have a person go and do the install. That was a lot easier before the pandemic. The pandemic, of course, made that difficult, but we're right now in the process of still catching up with donations that were made prior to COVID-19. This is a group in Ecuador. They do trypanosome analysis. Chagas is a big problem there, so they do toxicology on T. cruzi and use viability methods to run the little promastigotes through the instrument and determine if they are alive or dead. But we have a pretty good system worked out right now, and we have good relationships with freight forwarders and shippers to get our instruments in. We also try very hard to do training. ISAC has an education arm called the Live Education Task Force. Live Education Committee now, that organizes workshops all over the world. We bring in experts. The local organizers tell us what they want to learn, at what level they want, and we try to send in people tailored to that level. This is a workshop we did in Armenia about a year and a half ago. Maria Hymas, who's one of the founders of SciTech Biosciences, is there giving a talk about compensation and unmixing. Andrea Spittler is there. Ray Lanigan in the center, who's my partner in crime in finding machines and getting them restored, is there as well. So we try very hard to follow up these donations with workshops and training. We also try to support the maintenance. I and several others in our group have become reasonably experienced in maintaining the equipment. We've gone back and learned how to do it. But sometimes you can't travel somewhere. Right now, I can't travel to Russia, unfortunately. But we are still very much in touch with the group. We use Telegram and WhatsApp to do remote coaching to get the equipment running again. This is an interchange we had to try to get their sorter running last year. So it's a challenge, of course, but we're really trying to stay in contact with these groups and make sure that their equipment is supported. So as everyone in this room knows, flow is really a critical biomedical research for so many. It's a critical tool for what we do. And in some cases, in some of the places these machines go, there really aren't that many research cytometers. There are clinical machines, but they can't be used for research applications. We're also giving a second life to this equipment. Most of this equipment is still quite functional. It's not retired because it's broken. It's retired because the group using it needed greater capability. and had to move on to something better. But we're really trying not just to give instruments, but the technology of cytometry. We get the machine in there, we get it running, we make sure it's staffed, properly installed, we do our best to do the maintenance. We're trying very hard to provide reagent support, which, as everyone knows, is a difficult problem. And we try to organize educational programs for them. We don't just want to dump the equipment and say goodbye. We really, the long-term commitment is really, really critical here. And this is the best part of my day, when we get the email saying, this is from Gaia, saying the cytometer is in the lab. It is not on a table because we are waiting for more guys to place it. It's huge. So it's always a wonderful feeling when the stuff actually finally arrives in its destination. Now currently, because of all the organizational changes that are occurring where I live, we are changing our web presence. So I'm happy to share, if you want to get involved in this, to be a donor, to be a recipient, to be a technical advisor in a region that has a donated machine. For the moment, contact me, but I'll provide you with all the necessary web links and contacts. One of our big needs, and what we're really trying to build up right now, is to have local experts so that we don't have to fly people halfway around the world. And the local experts are here, so they can provide assistance, timely assistance to groups that are less experienced in cytometry. So I will stop. That's about 20 minutes. I'll stop there, and I guess I'm happy to take any questions if anybody has them. Yes. Yes, Kennedy. Here.
Thank you, Bill. Thank you for the great talk. I'm very, very excited about this, and I think it's something very critical that we establish the same sort of framework for establishing a flow cytometry center here in Kenya. And I was just wondering, could you highlight a little bit more on the procedures to get access to you so that you can provide some of the refurbished instruments to the institutions here who may need them? What do we need to do?
Absolutely. So right now, if you're interested, at this instant in time, the best thing is to contact me. We do have an application. Ray Lanigan, who's a major part of this, put together an application process where people can put in requests and if they have a particular need, and we'll consider it. We're pretty small right now. We're only doing a few instruments per year at the moment. We want to expand that. We have a major fundraising effort underway at the moment, so we're currently trying to expand our efforts. But if you contact me, I will send you the link for the application site. And also, if you know anybody who works in cytometry, we also, in fact, until recently, our main source of placements was referrals through ISAC members and other experienced cytometrists. So for example, Roxana, who is at Vermont, lobbied us long before we got that X20. She lobbied us for the CIDRZ group because they really needed an instrument. So she came to us and said, please, keep this on your radar. And when the machine came along, it turned out to be a perfect fit for them. For the moment, contact me because we're just completely revising our web presence. But I'll steer you in the right direction. Does that answer your question?
Great. Yes. Okay. I think if you need Bill's contact, you can get to me, or you can write directly to Bill. And I hope, Bill, you also provide online training sessions for people who want to be experts.
We will provide all of this. We work extensively with people like Hemen, who does extensive online training, and he is going to be your next speaker. But we're really trying to draw on the education resources of ISAC because we have wonderful teachers, members all over the world, and we're really trying to leverage that right now because we can not only get you help, we can probably get you local help. Hope that's not far away. Yes. Okay.
Thank you, Bill. That is a wonderful program. I really like the fact that you have incorporated maintenance support. Sometimes that turns out to be very expensive, yes, especially in fairly remote places. And so I was wondering if institutions are not necessarily even seeking for an instrument, but they have instruments, but they would like to be part of this network to get the other benefits like instrument support or any other. Are you able to accommodate groups that have other instruments?
Absolutely. I didn't discuss it today. We actually call it Restore in Place. It's a part of the program where if you already have a machine, we'll come and take a look at it and try to get it running again. I'm not an engineer, I'm an immunologist by training, but in the course of doing this work, I've been trained or trained myself. We're actually fortunate to have a number of retired service engineers who are part of this group who are a lot better at this than I am. So when we give a machine, we try very, very hard to provide the maintenance. Maintenance is prohibitively expensive, and in many cases, it's not accessible at all in some parts of the world, reasonably accessible. So that's part of our commitment when we give a machine, is really try to keep the thing running. But if you have an instrument already, and we've done that a few times just in the last week, we're happy to take a look at it. We can do PMs, we can change lasers, we can do all sorts of things. We've really tried to build up our repertoire of the service that we can provide. It's a lot easier for us if you already have the machine. We don't have to give you something. You've already got it. All we have to do is come and see if we can help you get it working. So by all means, get in touch with me about that. Definitely.
Okay.
Okay.
So thank you so much. I think I also want to just share with the team that Bill has been very kind. He came first to Kipre last year.
I think it was first time last year.
Yeah, the first time last year, and he actually supported us with maintenance of our equipment. I know I don't want to be mean, but he sacrifices his time to come here.
You're part of the.
But there's a way we can agree with him.
You're family now, so you.
So thank you so much. No, but mine was just to appreciate you,
Bill. We're going to do our best to take care of it.
Because it's a great sacrifice for you to just come and be here. Thank you.
No, thank you.
But you talked about the live. We could do these things live. I mean, you might be in the US, but there's a way you can support us online.
Yeah.
With maintenance. So I think that's also another way we can.
Yep, and he'll talk about that in detail. During the pandemic, of course, we all had to go remote, so we did an extensive remote training. Hement already knew how to do it. He had been doing it before the pandemic. We had to learn once that started. But typically our kind of path is we initially do a remote training just to assess interest and level of expertise, and then we try to organize something live. But the live education group at ISAC, which is separate from this, they organize workshops all over the world. We're going to do one in Mampong in Ghana in about a week and a half because they just received an instrument. And Sasha's going to be there. Andreas Spittler is going to be there. Rui Gardner, who's a really big flow guy at Memorial Sloan Kettering, he's coming as well. So, so we really try to organize these as a follow-up to donations.
Okay, so we appreciate you. Maybe another round of applause, kindly.
Thank you so much.
Okay, so we are just going to shift the program slightly. We have Professor Andrea Kozariza online. So he's a professor of pathology and immunology, serves in many capacities at the University of Modena, School of Medicine, Modena, Italy, and is also the past president of the International Society of Advancement of Cytometry that Bill mentioned. Let me give you a bit of his bio. Andrea holds a full professor of immunology in Modena University. He took a degree in medicine and surgery at the University of Padua, and then a PhD in oncology and a specialization in clinical pathology. His main scientific interests are immune changes during aging and longevity, the COVID-19 pandemic, HIV infection, sepsis, multiple sclerosis, bioenergetics and metabolism in lymphocytes, mitochondria during cell death, and in several pathologies, system biology. He has published over 450 papers in international journals and he's received greater than 55,000 citations. He was a president and past president of the International Society for Advancement of Cytometry. So Professor Kozariza, I hope you can hear us online.
Yes, sir, I'm here. Can you hear me?
Okay, we can hear you loud and clear. So thank you.
Okay, great.
Thank you.
Well, thank you, Lucy and Paul, for the kind invitation. I'm very sorry that I cannot come to Kenya, which is a country that I really love, as you can see from my first slide. If I can divide if I can share my screen. I cannot do this.
Just 1 minute, I think we're looking into it. Yeah,
Can you see my screen?
not yet. You've been given access to share.
Yes, I try again. I'm trying to share, but I cannot. This wants me to do something which I don't understand. I am required to change something in my computer to share the screen. Maybe I try to connect again.
Okay, please do. We'll wait for you.
I'm
Yes.
sorry.
No, you're okay, that's fine. As we wait, we're happy to take any other questions from the floor. Any question? There's one in front.
Yeah, the issue of donations of, I think, asking, all right, here with me. The issue of donations of equipment, sometimes from experience, although you said you follow up with training and maintenance, but over what period of time? And at some point that equipment has to be retired as well, right? How do you handle that?
That's a good question. I mean, we're a very small group, so we do our best. And you'll notice, for those of you who have been in flow for a while, some of these instruments are kind of old. They're fax calipers, which are wonderful instruments, but they haven't been manufactured in 10 years. So these instruments will have a lifetime. But I would actually give that lifetime, it can be 5 to 10 years more service. We do a lot of internal upgrades. We do them now before we ship the instrument so that you receive them that way. We're also very willing to train the local managers, core managers to do their own maintenance. Because this is not, again, I'm not an engineer. If I can do it, it's not that hard to do. So if groups are willing to do that's a very powerful thing. I think one thing though that's critical, and it's something I've learned as a core manager, is having a person who's in charge of the machine to maintain it is really, really critical. A core manager, someone who takes responsibility for it. We experience this all the time at my institute, where you'll have a postdoc or someone who runs it pretty well, but then they leave, and all that knowledge goes with them. And one thing that's great about Kipre is they do have a staff whose job is to maintain that equipment, QC it, make sure it's working properly, and having that continuity is critical. But you're absolutely right. We're still a small program. We can do our best, but we can't fly an engineer in on 48 hours notice. It's something that we'll just have to, we'll have to do our best with. We are now donating newer equipment. We've given an X20. We gave a Northern Lights, a SciTech Northern Lights that SciTech donated to us recently. They went to Argentina. So this is a brand new machine. We're also trying to work with the manufacturers. They're obviously not eager to get too involved with this, but I think that is changing. SciTech has been very helpful to us. They actually did the install in Salta, Argentina, which was very generous of them. So we're trying to improve this infrastructure. But you're absolutely right. Reagents are the other big problem. If you don't have reagents, you can't do your experiments. And we're still trying to solve that problem right now. So we're pushing to expand this program, but we're still in the early stages of it.
Allow me to, I'm just wary of time, so allow me, Hemant, to let you come on board as we sort of address. Okay, so today, again, I have the pleasure of inviting Dr. Hemant Agrawal, another very committed flow cytometrist. I think I met Hammond again the same time Bill was in Nairobi over a year ago. He is keen to come in and train within the space of flow cytometry. So when he heard we were having another workshop, specific at MKU, he offered to come back again. So I'm really happy to have you here again, Hammond. So let me just give a bio. He's the founding director of Flow Cytometry Solutions Unlimited in India. He's also an application support consultant, working with De Novo Software in the US, managing officer, the Trust for Education and Training Cytometry in India, and he's a member of the ISAC Live Education Delivery Task Force. His background is actually an immunologist, as I said, with a passion for flow cytometry. He earned his PhD in immunology from the University of Hospital Essen in Germany. And he began his career at the Oklahoma Medical Research Foundation, focusing on dendritic cells and macrophages using multicolor flow. He then moved on to the Northwestern Memorial Hospital in Chicago, where he continued working with research on autoimmunity. Between the years of 2011 and 2013, he served as an application and product manager for FlowJo in India, and really just spanned the whole of the Indian continent and Middle East. And he has authored numerous papers, but he currently is based in Jaipur, India, where he runs his flow cytometry solutions. And his passion is to train. So he's had over 85 on-site workshops, including now 2 in Kenya, and more than 40 online courses. So really, Hammond, it's a pleasure to have you back again in Nairobi, and we look forward to future collaborations. So I welcome you to give your presentation.
Thank you, Lucy, and the organizers for this workshop and the conference. It's really a pleasure to be here. And I'm so glad, actually, that I came and I interacted with the students and the faculty and the research people here. There's so much of potential in Kenya and as such in Africa that a lot can be done, actually. So in my next 20 minutes, I'll take you through that flow cytometry. I mean, we always think that this is just a technology. I can just run one sample, I get a one picture and done. But have you ever thought of that? Can you make a career out of that? I mean, there are many people, those who have made career, and one of the examples is in front of you in form of me, that after completing my postdoc in USA, I went back to India. I made a whole career in flow cytometry. So don't just think that this is just a technology that you use for running your samples and generating the data. A lot can be done with flow cytometry. So next 20 minutes, what I'm going to do, I will take you through what are the different options you can have and how you can become a good flow cytometrist. Okay, this is really important to be a good flow cytometrist. So first of all, that I'm not sure that everyone present here in this room is having a background in cell biology or flow cytometry. So it's really important to say that what is flow cytometry here. I think this is not, okay, it will not come on the screen. Anyways, no problem. So flow cytometry in short, we say it's a single cell analysis technology. And it's really powerful that you can analyze each and every cell. You can do it in any kind of a tissue, but what you require is a single cell suspension. So blood as such is a single cell suspension. But if you get samples like a lymph node, spleen or something, you need to convert that into a single cell suspension, and you can do wonders with this technology. And you generate these kind of a data. This is just a picture of a machine, and you get this kind of a data. Here on this data, you can see you can identify different cell types. And the beauty of this technology is that in a heterogeneous population, you can get your population of interest. And that is the really beauty. Like I have a different kind of a cells in a mixture, but if I want to just know about my cell, I can do it. And I need not to isolate it to do that. I just need to tag those cells with a specific antibodies against the specific markers, which are either presence over the cell surface or inside the cell, and I do those things. So it's a very powerful technology, and all these measurements, they are happening with a rapid pace, and you get a lot of information from this data. So the question which is all going in your mind might be at this junction that why this guy is standing here and talking about career and flow, because why flow cytometry? It's a very well-known and proven technology for cell analysis, utilized extensively in biological research and clinical diagnostics. It's an ever-evolving field. And you know, when you are associated with the ever-evolving field, you have always a chance to do something new. When I started my journey back in 2002 as a PhD student using flow cytometry, I used to do only a 2 laser 4 colors. And today, we have machines which can do 5 lasers more than 50 colors. So just in 20, 25 years, it has evolved so much. New things are coming every day. And there's a continuous requirement of that talented people. That's the key. That people are looking all over the world that I should get a good flow cytometers in my lab. And as Bill just mentioned, that we need a good core managers, a good core facility to run these machines, to maintain these machines. And it is promoted extensively all over the world by the users and the different societies. And as me and Bill are a part of the society known as International Society for Advancement of Cytometry, so we try to promote this technology over the world. Now, this is a very kind of a thing which I show to all the people in the field of science, that why you always look for alternative careers in science. The days are gone when I was thinking that I will finish a PhD and will get a job in the university. This was a commentary which was published in Nature back in 2011, which is 14 years back. And you can see the title is The PhD Factory, that the world is producing more PhD than we require. So where all these people will go? Because universities, institutions, they are not having enough space to absorb each and every one. So keep that thing in mind and think about it, that how you have to decide your career. And flow cytometry is one of those things. And the challenges that we have in front of us today, that the scientific funding and the number of job positions are decreasing today. It's not like that which was there in last decade. And young scientists must be prepared for these career challenges. Because I don't want you to land up that you completed your PhD and postdoc, you are still looking for a job. So why not to make yourself, why not to create yourself in such a way that as soon as you are out from the institution, somebody is waiting for you with a job on the plate. But for that, you need to really work hard. So flow cytometry rocks. This is something where you can come. Why? It's high in demand. Everyone wants to be a flow cytometrist. Today, if you see any ad coming from any big lab, they say, do you have experience in flow? Can you do the sorting? Can you look at the different cell types using the flow? If you have that, yes, that's the job waiting for you. Having exciting career paths and stand out in opportunities. A person, if you're with experience in flow, it's always hired first. Especially I'm talking about immunobiology and cell biology. And of course, when I'm doing a job, I also look for some money. So you have an opportunity to get a good amount of money. Also as a technologist or a core manager or a scientist, we're using flow cytometry. And why you should be excited about it? Because you are the one who's going to shape the career of future medicine. Okay, so keep this thing in mind and think about it if you can have a career in flow cytometry. And now comes the numbers. The numbers are really important to understand what I am talking about, the seriousness behind that. So if you look at the market growth and the industry demand, market size of flow cytometry by 2032, it's going to be $7.7 billion. And the annual growth is about 11%. So the market is growing like anything. And it is having a numerous application in research, in clinical, in industrial applications. So everywhere you have the options, you can go. But what you need to learn this technology in a right way. So next thing that I want to talk a little bit about this data, that if you look at the applications of flow cytometry by demand, then you can see it is having research, clinical, industrial, diagnostics, and the drug discovery. And every year it is increasing. So this is the data from 2024 to 2025. And then these new things which are cell and gene therapy and drug development is always there. There's a lot of requirement of flow cytometry. I don't know how many of you were there in the lecture, plenary lecture given by the director from IVI Seoul. He was talking about from A to Z, that how the vaccine development happens. And flow cytometry plays a major role in preclinical vaccine development. So there also you have a lot of potential to be hired as a flow cytometrist. And if I go a little bit further, if I look at just as cell and gene therapy growth, then over the years you can see the growth is increasing. And if I look at the immunotherapy market, again it is increasing. If I look at the job sectors in different academia, biotech, CROs, pharma, the growth is immense. There's a lot of growth which is happening. And then if I look at the salary structures, you can see with every year the salary is also increasing in these areas. So I have something which can give me a good salary, which can give me a good recognition, then why not? Why not I can make a career in flow cytometry? So what are the different careers I can have? So next 2 slides, I will talk about how you can be hired in different labs and what kind of a career you can have. You can be a research scientist in the lab. That's academia or non-academia. You can be a clinical specialist, starting from technologist to the person who reports. You can be anything. And you can also join any company as a biomanufacturing technician or a scientist. But if I divide this into greater details and try to see what are the different options that I can have as a flow cytometrist, then I can see I can be at the academic research labs. I can be at the pharmaceutical biotech companies, working as a cytometry core scientist, principal investigator, or technologist. I can be at the clinical diagnostic lab. Or I can run the whole core facilities. The core facility managers, if you look at their profile, they are cell biologists, immunologists, and now they are helping the other labs to run their samples in a correct way, to help in correct way of their data analysis. So the lot is happening in the field of flow. And you can be a part of the instrument manufacturers also. As you can see that our friends from Beckman Coulter, they are here. So there's a lot of jobs which are there with the companies also. You can be application specialist, you can be field service engineer, you can work with them as in R&D, you can be a product manager, just talking about their products. Or you can join the government agencies. Like for example, in US we call CDC, NIH, or FDA, etc. In your Ministry of Health or anywhere else, you can join as a flow cytometrist. And you can be a data science analyst. This is again, I want to give a half a minute on this. Data science, it's going to change the future. So when I said I started my journey with a 2 laser, 4 colors, and now I am on 5 laser, 50 colors. So do you think the complexity of data remains the same? No, the answer is no. Today, the data is highly complex. You cannot just analyze the data with a 2D plot. Those days are gone. So you are looking for something we call high-dimensional algorithms. So you can make a whole career if you know the flow the right way. So you can become a data analyst. These are some very good options, and these are remote jobs. You need not to go to the lab or to your institution or to your company every day. You can work from home. So these are some very important things that are happening in the field of flow. And you can be at the regulatory affairs department of the government, of the institute, and can get options to work with them. So now let's come to how much they are going to pay you. These salaries that I wrote it down, this is as per the standards of the developed nations like US and European countries, but you can think about like you are in the in a bunch of people, those who always will get a higher salary than your peers if you are a flow cytometrist. So if you see that you have an entry level where you start something with 45K to 50K, 60K, then you go to the mid-level when you have experience of 2 to 5 years, then 5 to 10 years, and then when you have a 10 plus years experience. So with more the experience you gain, the more the salary increases. So you have the salary also, good salary, and you have a good field to work into it. And then, sorry. So if you see, this is 85%, if you look at this data, it is more than 85% above the average biotech salary. And if you look at this, you're in the top 25%. So this, I think this should motivate you to make a career in flow. That your knowledge can give you a good job, which can pay you handsomely. So now, as a student, what do you need to do? You need to do SWOT analysis. That you want to know why I need to be here. Because your strength, the current strength, that you are a cell biologist or immunologist or microbiologist, aligns with this field. You can have a career transition, overcome traditional academic limitations with a specialized technical skills. There's a lot of market opportunities, and you have a competitive edge over the other people in the same field. So why flow cytometry beats the traditional path? If you look at this pie chart, that flow cytometry career, it's like a lot of people want to choose this one as compared to traditional academia or journal lab tech or the other biology labs, because it's a highly paid field and ever evolving. A lot is happening in this field. So how I can get started? What I need to develop in myself to be a flow cytometrist, to be hired by anyone? You take courses, you do courses, good courses. You learn, and learn. As much as you learn, the more you grow. You seek lab experience, become a volunteer, do internship, research assistant, go to the labs which are into flow, learn as much as you can. Attend conferences, workshop. As you can see, you all are here. We are talking about flow. Some people might be at the workshop that we did at MKU. So you learned something there. So these are the experiences that you are gaining. Then always read good scientific literature. Reading and reading and reading. It will really help you in developing your career. Networking. I always believe in network. And networking is very, very important. Never hesitate to shake a hand with the people. Never hesitate to share your cards. Talk to the people and say, okay, I am this and this, and I am interested in your work. Can we talk a little bit? The more you talk, the more you network, you have a higher opportunity to be hired by the people. Because the thing is that now in today's world, we always want to hire someone that we know. As you know, like unknown person is better than the known person. Maybe that person's the devil. It doesn't matter for a moment. At least I know that person. And then explore some online resources. Because many a times you are not able to go physically to these workshops and the training programs. So look for the online resources. But learn from the right resources. That's the key, not from the bad resources. So essential qualification that you want. You need to have a technical competencies and educational pathways. As I talked about, you should have a bachelor's degree or a master's, depending on what level you want to make your entry. So accordingly, you should have a degree. There's a lot of jobs which are available as a flow cytometry operators. For that, you don't require a PhD. Maybe a master's or a bachelor's is more than enough. Even for core managers also in US, there are many core facilities where they are not run by the PhDs. They are run by the good people, those who are with a good master degree. But what do you require at the end of the day? A good knowledge in the field. So how to get this? The training and the certificate pathways. So you should take the courses, online, offline, specialized training. For example, we have this training coming up this month, starting 22nd of November, where we are going to teach people about how to do this analysis in a correct way. So that is a 1-month course. So if you take that, your level of understanding of flow cytometry will go to a next level. So if you guys are interested, please visit our website, flowsalts.com. And you can have that look at the program and see if that interests you, and if you want to train with us. And get some practical experience. Find the place where the machine is, get the practical experience. So last 2 slides. It's a future trend, which is happening here. High parameter flow is happening. Automation and miniaturization is happening. As Bill was showing you the big machines, and now we have machines of this size. So things are changing. Imaging flow cytometry is there. AI is there. ML is there. And then you can also utilize at the point of care diagnosis. So this is a quote from the industry leaders. So flow cytometry is just, it's not just a technique, believe me. It's your gateway to transforming patient lives through cutting-edge science and technology. Keep that thing in mind. It's not just one picture in your paper. So the conclusion is flow cytometry stands at the intersection of biology, technology, and analytics. It offers a dynamic, intellectually rewarding career path with global opportunities. Every cell tells a story. So learn how to listen. Okay? So you have to go ahead and think about this. So thank you so much for your attention. And I'll be very happy to take your questions, if not now, maybe later on. My details are here. So feel free to contact me if you want to know more about what kind of a career options you can have as per your degree, as per your educational background. So thank you so much for listening to me. Thank you.
Okay, thank you so much. So Andrea's online. Please allow us to let him.
Yes, sir. Hello, sorry for the problems. I hope now it works.
Okay, just give me a minute, Andrea. So we'll allow him to speak and then we'll take questions at the end. He says he has to leave by 10 minutes past the hour. Okay, so please save your questions.
Yes, okay.
So,
okay, can
Andrea,
you see
go ahead and
it?
talk.
Can you see the leopard? Can you see my picture?
Yes, now we can see. So please go ahead and present. Thank you.
Okay. Well, thank you again for the invitation to Lucy and Paul. It's a pleasure to be here. So what I will talk about is the role of immune response in different situations, especially when the immune system is suppressive. It's suppressed by external agents like drugs or viruses. So I will talk about when a strong immune response is immune suppressive, even if strong, immune response to Mpox in persons living with HIV, and I will mention the response to the vaccine for SARS-CoV-2 in patients who have multiple sclerosis and take immunosuppressive drugs. So when the strong immune response is immune suppressive, so in the case is very well known, is the case of COVID-19, in which in the initial phases of the infection, we have a very strong immune response, and as you can see here, a number of cells like CD3, CD4, CD8, and so on are under strong activation, so they are activated, but in the same time, they are also suppressed because they are not able to perform properly their job. And even worse, in some cases, it happens that these changes are so strong that most CD4 cells in this case, or CD8 in another case, do express markers of senescence or even apoptosis. More than that, we have seen years ago that when in cells are highly stimulated, they can produce a huge amount of cytokines, many of which can be detrimental for the patients, as in the case of interleukin-17, which is highly produced by patients with COVID-19 and activates neutrophils that are able to go to the lung and induce damages. So this is a typical example of what a strong immune stimulation can produce, which is paradoxically a strong immune suppression, or in any case, a bad situation for the patient. What about CD8 cells? As in the case of viral infections, also CD8 cells are highly activated, but as you can see here, with this 20 color flow cytometry, what came out is that such cells do express markers of activation, senescence, and apoptosis in the same moment. It means that they would like to work, they are activated, but after activation, they do not exert their job and tend to die. So we put all the data of the immune response in a framework, and the results, we summarize what was found by our group and others in a paper in the Lancet Respiratory Medicine showing the importance of CD8 and CD4 activation, here showing the fact that the neutrophils goes up, that Tregs go down and go up again. B lymphocytes became plasmablasts and are not able to produce good antibodies anymore. And monocytes are activated as well as other cells. So to make a long story short, what came out is that if you activate too much the immune system, you have an immunosuppression. The second case of in which I will mention is the immune response to Mpox in persons living with HIV. Mpox is now defined as an AIDS-defining event. It is not only an infection due to Mpox, but also something that can occur in persons living with HIV, and very recently became one of the most important AIDS-defining events in the sense that many other events are no longer present, like pneumocystis carini or so on, because the drugs work very well. But in this case, Mpox is quite important. So we were interested in understanding whether Mpox the response to Mpox was efficient in persons living with HIV, and we take the opportunity to study patients who get the infection after a trip to Africa, and we know that in Africa, Mpox is a problem and has been a problem for many years, and especially in West Africa, as you can see here. And we could study people who came from that area and developed Mpox. So the question was, what happens to the immune system? Which is the first response to Mpox? And the data were published in the Lancet Ideas 2 years ago, 3 years ago. And the question was, we can see an immune response in healthy individuals infected with Mpox. But what happens if the infection occurs in patients affected also by HIV? So the question is, can patients with HIV respond to a new virus? Actually, we call Mpox, no monkeypox. Okay, what came out? First of all, we saw that in the blood of patients with Mpox, there was a decrease in the number of CD4 cells and an increase in the number of CD8 cells. More than that, different populations had different behaviors. And again, what came out is that even if the number of CD4 cells were low, the amount of effector memory and central memory were higher in patients with monkeypox, as well as in CD8 T cells, where we saw a marked activation of such cells, and the activation of the cells identified by the expression of CD38 could decrease with time, either in CD4 and in CD8. Again, other markers, like markers of cell exhaustion, were highly present in patients with Mpox, but fortunately, they went down with time. So, a more detailed analysis of CD4 and CD8 with UMAP bidimensional plot confirmed what we have seen by conventional methods. This is, as you have heard before, a new approach to analyze data in an unsupervised manner, and comparing unsupervised with supervised data, it came out that the results were fortunately the same, in the sense that we have observed in CD4 a significant increase of memory activated cells, residual memory cells, of TEMRA cells, and so on, and in CD8 even more than that. The response was able to persist for some days, and in fact we have seen that if you analyze the amount of cells responding to the viral peptides by the use of the test detecting interferon gamma, you can see that these patients, all patients, were able to respond to the peptides producing interferon gamma. And this was true for all individuals of the group, and in fact, among the 17 patients that we could study, 10 were HIV positive and 10 were HIV negative and 7 HIV positive, and all of them responded perfectly to the virus in vivo and to the viral peptides in vitro, showing that the infection with HIV in patients who are positive but who take the therapy and are also very well virologically suppressed. They do not have HIV viral load in plasma, they have a decent number of CD4 cells. There are no difference in the response to a new virus like mpox. In other words, if you have HIV and if you take the therapy, if your immune system is still working, you can get a new infection, and a new infection is fought exactly how you were HIV negative. So that's a good news for all patients who are in therapy. What about the response to vaccine in patients who are immunosuppressed for different reasons? What we did in the last 2 or 3 years was to study patients with multiple sclerosis under different treatments. You know that multiple sclerosis is an autoimmune disorder in which you have a number of modifications of the immune system, but basically what you produce are antibodies or T cells against the myelin of the central nervous system, according to which you undergo serious problems in a few years. So now we have a number of drugs that are able to block the course of the disease at different levels. But you see here a cartoon showing how many drugs you can have that intervene at the level of the egress of lymphocytes from lymph node, at the level of antibody production, at the level of contact of the macrophages and lymphocytes and the microglia, and so on. So we have a number of different drugs. So what we did was to take a cohort of about 100 and some patients, 106 persons, among which 94 were patients with MS, who were taking different drugs. You see here cladribine, the fumarate, fingolimod, interferon, natalizumab, teriflunomab, ocrelizumab, and so on. So they were taking different drugs. So the question was, what happens if a patient who is severely immunosuppressed because of the autoimmune disease received a vaccine? Do patients respond to the vaccine or not? That was the question. So we took, again, patients with different therapies, and we tried to detect the T cell numbers and the response to the vaccine. So the first thing is that, in principle, the quality of the T cells, apart in one case, in the case here that you see with fingolimod, apart this case, patients with fingolimod, the number and the percentage of T cells were not so different from that of healthy individuals. And more than that, analyzing different T cells population, effector memory, central memory, transitional memory, and so on, you see here different drugs, and the changes were very little, if any. Just in the case of teriflunomide, we saw a change in the transitional memory cells, while patients assuming other drugs did not show relevant modifications of the T cells in the periphery. But the question was, okay, fine, but what happens if I want to detect and to study antigen-specific CD4 T cells in these patients? I remind that all of these patients were vaccinated 3 times for the COVID-19. And so what we did was to take the cells after the 3rd vaccination, 1 month after the 3rd vaccination, and to cope cells in vitro with viral peptides to see the specific response to the vaccine. Again, to make a long story short, the quantity of the T cell response in terms of production of any cytokine was not dramatically different among different patients. Only those taking fingolimod, again, show a higher production of granzyme B and a low production, if any, of interleukin-2. So the point is, they do have a similar response in terms of quantity of cells, but a different response in terms of quality. Same thing happened for the CD8 T cells. Also in this case, fingolimod have some main effects on the percentage of CD8 T cells and also on the quality of the T cells in the blood. But what happens to antigen-specific T cells? Again, also in this case, the amount of responsive cells were more or less equal in all groups, but those taking the drug called fingolimod had a very high percentage of T cells, antigen-specific T cells, able to produce granzyme B more than other cytokines. So what about B cells? B cell, we did a similar analysis. Amount of B cells was more or less similar, but patients taking this drug here, I mean the fingolimod again, fingolimod is here, this cladribine, they did have a consistent change, and those patients taking ocrelizumab or taking rituximab, which are antibodies that recognize B lymphocytes and deplete B lymphocytes in patients, those taking this drug did show a negligible amount of B cells as expected. So the point is, but what happens to these patients from the point of view of the immune response to a specific antigen? Again, we measured the production of antibodies in these patients, and what came out is that those patients taking drugs like rituximab or ocrelizumab that deplete B lymphocytes, they did not show a response in terms of antibody production, but they had a very good response in terms of T cell production, antigen-specific T cells. So the point is, they do not produce antibodies, but they are very well protected by T lymphocytes, because none of them develop COVID-19 after vaccination, in the year after vaccination. The last slide are very quickly about what we tried to do to understand what was going on. So we could perform by using a new assay that we have developed by mass cytometry with 44 markers or something like that. We have developed a single cell metabolic profiling analyzing different metabolic pathways like pentose phosphate, glycolysis, amino acid metabolism, fatty acid metabolism, mitochondrial dynamics, cell biosynthesis of DNA and 3 carboxylic acids. Then we have marker for signaling and transcription all together in one single panel, which is represented here. There are 22 phenotypic markers, 21 metabolic markers, and 2 cell quality markers detecting a lot of pathways in the cell. And what we found was that analyzing such markers, we had the possibility to understand another thing, that is how cell metabolism can influence the response to an antigen. And we have done that also by using a different method, which is called SHEMIT, that is a sort of movie analyzing the capacity of cells to depend on glucose or to produce fatty acid oxidation or to depend on mitochondria or to have glycolytic capacity. So the story is that antigen-specific T cells were found among CD4 or CD8 cells, and the test is called SHEMIT, first of all. Here you see the antigen-specific cells among lymphocytes, T cells, among all the other subtypes. So the story, again, is that patients assuming a given drug, like in this case interferon gamma, had a low capacity to oxidate glucose, and they had a low glucose dependence of both in CD4 and CD8. About other cells, we could define different metabolic states in different cell populations. But what is most important, in my opinion, is the fact that according to the change that you see in the periphery, according to what you have seen among CD4 and CD8, you can cluster patients in terms of the metabolism, and we could detect different metabolic states according to the combination of different metabolic markers. And as you can see here, patients taking drugs like fingolimod or natalizumab, they cluster in different areas of the principal component space, showing that what they do in terms of metabolism is strongly affecting the quality of the response. And more than that, if you perform an assay, a prediction assay like this one with pencil methodology that allows you to understand what happens in the last year, we did a very simple assay. I mean, we took the data and we followed the patient for 1 year to see which patients get the infection after vaccination. And what came out is that patients with some markers did develop a breakthrough infection after vaccination in the year after vaccination, while patients with other markers did not. So it means that in our case, metabolism is very important to drive the quality of the immune response. And in fact, to summarize these results, the most important thing is that phenotype, function, and metabolism of antigen-specific T cells and B cells in patients with multiple sclerosis treated with different drugs mimics in some way the characteristic of the age immune system, because they are highly activated and they do respond in a different way, producing different cytokines. We have seen that some drugs are able to change the pattern of the production of T cells and B cells, and we have seen that we can predict some results using metabolic markers in the year following infection. So I would like to acknowledge a number of people in my clinics, in the infectious disease clinics, in the intensive care, and in the laboratory of clinical pathology. And of course, I want to thank you for your attention. And again, I apologize for not being here.
We want to say thank you very much. Thank you very much. I hope you can still hear us. Right? Are you still there?
Yeah, sure.
Okay. Do we have time?
I'm here. Yeah.
Do we still have time to ask you questions, or?
Yeah, sure.
Okay. All right. So I think I want to open up the floor for questions. I think we'll start with Andrea, and then we can go to Hammond. I'm happy to take questions. So let me just think through the question. There's the monoclonal antibody, the natalizumab you mentioned. I just want to understand a little bit more in terms of, we look at immunotherapies and the use of antibodies, but how, based on the data you presented, can you just give us an insight in terms of, you have your healthy controls, you have the ones that are sick, and you're treating with the antibodies. For those of us who are thinking around the space of doing treatments in patients, what would be the appropriate markers, and what would be, what can you say would work, and what doesn't work?
What does not work, you mean?
In terms of...
Well, this is difficult.
Yeah, because you've got so much variation when you look at some of these parameters. So maybe you can just guide us with the example you gave on the natalizumab experiment.
Yeah, natalizumab is a drug which is very potent. It is able to block the entrance of immune cells in the central nervous system, but it does not change what happens in the periphery. So it depends on which is your immune status, and independent on that, you can start therapy. And what I know from my colleagues in the neurology department is that you choose the therapy according to the age of the patient, to the status of the patient, and to the capacity of the patient to cope with side effects that every drug is. So in this moment, there are drug treatments, very, very potent treatments, and patients can live 10, 20 years free of the disease, free of symptoms. The point is that you have to choose the therapy according to the age and to the side effect that you have to avoid. For example, if you use antibodies for B cells, like rituximab, like ocrelizumab, you deplete completely B cells, and you have to be careful because you can get viral infections or bacterial infection, but you use these drugs only in patients who are severely compromised. Those patients who are not severely compromised, who have just very mild symptoms, and the patients who have the form that we call relapsing remitting form, in which you have some attacks of the disease and long periods without the disease, in those patients you can choose a drug which is more flexible, lighter, like natalizumab in our case, because you just block the ingress, the entrance of immune cells to the immune system, but you do not block an efficient immune response. So you have to modulate the response. But what I liked of this study is the fact that we could show that even if you treat patients with immune-suppressant drug, they do respond to a vaccine, which is very important for the health of the patient. And in fact, those patients who developed the infection after the treatment, after the vaccination, but they continued to be treated, they did not have a serious form of the infection. They just developed a mild COVID and have no problems in coping with this.
Thank you. Any other question? Just give me, there's one question for you at the back.
Thank you very much for a good presentation, Dr. Andrea. From your presentation, I've seen that you are comparing the mpox immune response in HIV positive and HIV negative individuals. In terms of their immune profile, that is in the CD4 count of the sick people and the negative people, how were you able to compare them? Now, in terms of their CD4 count, that is, if they were immunocompromised, to put it in other words, how healthy were the healthy people and how sick were the sick people, that is HIV positive? And
thank you.
What was maybe these sick people, how were they on ARVs? And in any case, they were on ARVs. Was ARVs affecting the mpox immune response as well? And the last one, you played a map of Africa, and I realized that there was a region in West Africa separated by just a country, and the other side there were mpox, this side no mpox. What could be the cause of that we see in the spread of the disease? In any case, your team is also involved in surveillance. Thank you.
Okay, you have a lot of questions. So the first is healthy people were very healthy because we use a age and sex match cohort of donors who are usually our colleagues or nurses or students, so they were matched for age and sex. And on the contrary, also patients with HIV, they did have HIV, but they were healthy because they were taking antiretroviral drugs and they show no symptoms of the disease, so they were fine. So from that point of view, the population were very similar. The number of CD4, of course, were lower in patients with HIV, but I would say all of them were over 800 cells per microliters, so the immune system was efficient. About that, there are no many doubts. The quality of the response was the same. The number of CD4 cells were less, but the amount of cells responding and how they do respond was similar. The point is that we could not find, of course, a response to the virus, to the viral peptide, to mpox peptides, in patients, in people, in healthy people who had no infection. Because if you don't have an infection, you do not develop antibodies or T cells. So the response was only made in patients with the infection. Those who were HIV positive responded similarly to those who were HIV negative among this group. So healthy donors were used for comparing the absolute number of T cells and CD4 cells, but among the population that we have studied, patients with mpox, those who had HIV or those who were not were similar. About the other question, what about Africa? Actually, I don't know why, but this was the outbreak of smallpox, mpox in the years from 1971 to 2019, if I remember well. So this was the number of cases that came out in that period. And honestly, I have no idea why countries like Kenya, Ethiopia, and Somalia were not affected in comparison with Nigeria, Congo, Cameroon, and Gabon, and Cote d'Ivoire, and so on. There should be maybe some geographical change or some geographical reasons according to which the infection could not be spread among the different countries.
Okay, so Andrea, we want to just say thank you very much. We were very happy to hear about your multiple sclerosis, mpox, and HIV. I think the question on the mpox, the different clades, There's clade 1 and clade 2, and now there's a clade, is it 1B? So it's a similar story with HIV. You know, we get it worse in East Africa, but the West African is not as bad. But that was a good question. Thank you so much. So if there's no further questions, I'm weary of time. So we want to just say thank you again. We can just again applaud our speaker. We hope you will find time one day to come back to Kenya. It's been a while. So thank you very much, Andrea. All right. So we'll now move on to our next speaker. I think Haman's still here. And if there are any questions, please, you reach out to him. I would like to now invite our next speaker, Dr. Margaret Mbuchi. She's a principal research scientist at the Kenya Medical Research Institute here in Nairobi. I think the beauty about Dr. Mbuchi is she was invited by the Excella team, Beckman Coulter, and she's been doing what we call a multi-center study, look at immune response status within visceral leishmania. So we look forward to your presentation. Welcome.
Thank you, Dr. Ochora. Thank you, everyone. So as she has said, my name's Margaret Mbuchi. I'm a principal research scientist at the Kenya Medical Research Institute. By way of a quick introduction.
Sorry, Doc, let me just make an announcement. I forgot to say something. Dr. Ruth, if you can just stand up. Sorry for this. She's kindly requesting all poster presenters to step out and see her. She's there at the end, so please go and see her. Thank you. Sorry for that, Doc.
Okay. So by way of a quick introduction, as I said, I'm a principal researcher at the Kenya Medical Research Institute. I've worked at KEMRI for many years, but I really, in the context of this presentation, my experience in research has largely been in the field of human visceral leishmaniasis, where I have worked for the last more than 20 years. And I've been mainly involved in diagnostics, development, and validation. But recently, I got an opportunity to collaborate with a few some partners from the East Africa region as part of what I'm going to present today. And I hope I can. So my title is rather mouthful, and it's a KEMRI focus of a multi-center study to assess immune response status in patients before and after treatment for human visceral leishmaniasis. And I would like to share our challenges and triumphs of establishing flow cytometry centers of excellence, because this was one of the key objectives of this project. Sorry. Oops. Oh, dear. Oh, my. Ooh. I think I'm a bit, I don't know whether I'm going back. Okay. Oh, my goodness. Sorry? There? Sorry. Oh, it's on? Move forward. Forward?
Yeah, and to backward.
Oh, okay. So back. So by way of a quick background, I said this project was in the context of human visceral leishmaniasis. So human leishmaniasis, as we know, most of you know, is a vector-borne disease, and it is one of the very neglected tropical diseases, and it's caused by an array of species of the obligate intracellular parasite in leishmania, and there are various forms of leishmaniasis, including visceral leishmaniasis, which is the most severe, mucocutaneous leishmaniasis, and cutaneous leishmaniasis. And the visceral leishmaniasis is the most severe form, as it affects spleen, liver, and is fatal if left untreated. And the East African region is currently harboring the highest burden of this disease globally. And to address ourselves to that, as the East Africa region, last year, a strategic framework for the elimination of this disease was launched. covering the strategy between 2023 to 2030 in terms of elimination approach and what we need to do. Now, treated visceral leishmaniasis patients will normally go on to develop a unique skin form of the disease, which is known as post-Kala-Azar dermal leishmaniasis. And this disease, this form of human visceral leishmaniasis is known to serve as a reservoir, at least in our region, and it is therefore thought to play a key and very important role when we talk of elimination. And it is in this context that the East African region, 4 countries where this disease is highly endemic, that is Uganda, Kenya, Uganda, Ethiopia, and Sudan, became a part of what is called the PrEP-PKDL project that I'm going to talk about. Now, without belaboring, the PKDL disease, as I have already said, it has implications because it affects the face, the trunk, and it can be very stigmatizing. I won't go into all that because I've already mentioned. Now, I want to go to the second last statement. It is not known why PKDL develops at different rates with different frequencies among the patients in the East Africa region. For example, while it is assumed and known that visceral leishmaniasis in the East Africa region is caused by Leishmania donovani, when you go to the 4 East African countries, Kenya, Uganda, Ethiopia, and Sudan, you find that in Sudan, upwards of 50% of patients who are treated go on to develop the PKDL form of the disease, whereas those numbers are very few in Kenya, Uganda, and Ethiopia. And so this consortium sought to address itself. So some of the things that have been postulated is this a feature of the human genetics? Is it a feature of the genotype of the parasite in circulation? And in the context of the work that we were going to do, is it a feature of interaction of the parasite genetics and the host immune response? And so this is what the PrEP-PKDL project sought to address itself to. So very quickly, the primary objective was to assess skin immune responses, parasite load in patients before and after treatment. And then the secondary objectives was to identify systemic and local immune correlates of treatment outcomes, to evaluate parasite genotype in relation to immune response characteristics. And so, and to compare immunological and parasitological data across each of the East African study sites, partner institutions, and determine possible correlates of progression to PKDL. So we wanted to understand, to look at the parasites from the patients within the East Africa region, and also characterize the immune responses, both systemically, but also the skin, from the skin. And I just want to mention that the outcomes here, which are relevant to the flow cytometry and what I'm presenting today, so data on immune cell phenotypes and function in peripheral blood and tissues, and we wanted to determine, to investigate this using flow cytometry and other mechanisms or other approaches to understand what is happening. So very quickly, the PrEP-PKDL project was a large project, by the way, and one of the main focus of this project was to actually do a vaccine trial in Sudan. So Kenya was not involved in that part of the larger project. And then we saw an opportunity as the East African region to characterize the immune response and to look at the post-immune response parasite genotype interactions that potentially could explain whatever observations were made as a feature of the vaccine evaluation. But one of the major aims of this project was to enhance immunology research capacity in East Africa by coming up with centers of excellence for flow cytometry. And So I'm going to limit myself now to the smaller study, which is what we at KEMRI implemented, which was dubbed Immune Start at Cure, to look at the immune response characterization, the parasite genotype before and after treatment. I don't think I want to yeah, okay. Oh yeah, okay. So I just want to say that we focus, our aim was to look at the VL cases before treatment and after treatment, but also compare with healthy blood donors. And I won't go into the exclusion-inclusion criteria. So I want to now focus very quickly on the main focus of this presentation in the context of this conference. So we received, each participating institution received this Cytoflex LX from Beckman Coulter, and you can see it's a very, so we received this very advanced Cytoflex to do our analysis, and each participating institution received the same identical equipment. Then we went on to have a training for flow cytometry managers that were appointed or hired by every research institution. And the training was done at the University of York, who was the coordinating institution. And here you can see that with this Cytoflex LX, we were not only able to do to characterize the immune response using whole blood, but also we could go on to use it to do bead array, bead array for cytokine analysis. Wow. Should I face that way or where? Oh gosh. And as part of this exercise, we required a very well organized training, and we needed to move in tandem because some of the reagents we used were actually custom made. So the antibodies we were using were custom made, and so we needed to have a very synchronized trainings and to move in tandem with each of the research institutions. And this is a slide that I'm just using to show that we needed somebody, an expert who came to help us establish. And by this slide, Professor Harman said he has worked with Karen Hope. I just borrowed a slide from her presentation. This is one of a slide showing the first time we enrolled the first participant who was a healthy control, and that helped us to optimize the processes. So then we went on to do isolation, culture, and sequencing of clinical isolates of Leishmania. And we partnered with a team from the University of York. And here I'm showing a slide where we had a team from the University of York come to train and to actually do whole genome sequencing of parasites at KEMRI using the Oxford Mini Ion sequencing device. So very quickly, this was the journey that our study needed to take. And this study, by the way, was done 300 kilometers from KEMRI headquarters. So we did this study at the Chemoligot Sub-County Hospital, and we had to move our samples. Within 18 hours, they needed to get to the flow cytometry lab and be analyzed immediately. So we had to have a very well-organized system of moving of samples, sample reception analysis, and also have featured here the fact that some of the analysis will be done at our partnering institutions abroad, and so we needed to have a very well synchronized plan and strategy to be able to succeed. So what are some of the challenges that we encountered? This project was funded by EDCTP, and the start date was April 2018, and we were caught up in the COVID-19, and we had major delays. For example, our Cytoflex was held up at Heathrow Airport for 18 months, And remember I said we needed to all start together and move in tandem. So these are some of the challenges. Delays due to COVID-19, some challenges with procuring supplies, but for Kenya we were very happy and lucky because the Exila group really helped us and we were able to get our supplies. Some of the other challenges include visas and for example, our floor manager we had appointed could not get a visa to go to the UK, and the person that we hired later on got a green card, and as has been said, I think when you have flow cytometry training, that really helped her to get a job in the US, just the exposure to our study. So the other challenges were to do with compliance with Nagoya Protocol. I don't know how many of you are familiar with that, and as I speak, we are still trying to resolve that, which affects our ability to share data and even to publish. I think then the other challenge was international conflict. During the lifetime of this project, war broke out in Sudan. I know I'm focusing on the Kenyan side, but war broke out in Sudan where the larger part of the study was to be done. Another war broke out in Ethiopia, and during the time of our study, we had to navigate through dawn to dusk curfews that affected Baringo. So I think I just want to say that these challenges, we were able to do, to apply the approach that Haman said. The trainings that they are doing, this is how our staff were trained, and we had a lot of remote interactions for us to succeed. And I won't go through that. So in conclusion, overall, despite the many challenges, we were actually able to meet our targets. And actually, Kenya did a very good job. We were told by our coordinating institutions that we actually redeemed the whole consortium. But that can be understood because of the conflicts that came up in Sudan and Ethiopia. And we are very pleased that we were able to succeed. Now, I just want to quickly mention, so yes, we built centers of excellence. However, very quickly and relevant for these discussions, especially with the foregoing presentations, is that, so post-project, the need to have a post-project continuity to use the equipment. This equipment budget alone was 250,000 euros. So we have this very fancy equipment sitting in our lab, and the reason I actually requested to come to this meeting was to actually network with people that we can partner with. So, but despite the challenging circumstances, we were able to achieve our targets, and we are very grateful. And I'm very happy to have met the teams here, and especially the ISAC affiliated teams, and also to hear that there are people, we can have donations, but also get some help when we need, like, to maintain our equipment. So the last slide here, I say that all is well that ends well. So that was the first meeting, kickoff meeting in 2019, and then we closed this project in the month of March this year. And I want to acknowledge, first of all, our study participants, the Chemoligot Sub-County Hospital, the University of York, and all the partners within the PrEP-PKDL consortium, and the Kenya Medical Research Institute and the study team. Thank you very much.
So much, Dr. Mbuchi. We would request we go on to the next presentation, and we'll take the questions at the end. Thank you once again. The next speaker is Dr. Daniel Mwema. He's a senior research fellow at the CAV Institute of Clinical Research and a lecturer in the Department of Medical University at the University of Nairobi. His research focuses on several areas: cellular, genetic, molecular mechanisms, underlying immune responses to infectious diseases and vaccines, and a wide area of many other things. So, Dr. Mwema, nice to see you. We look forward to your presentation. Thanks.
Thank you, Lucy.
I'd seek you keep to time. We've got 2 more left.
Yes.
Yes, thank you.
Thank you, Lucy, for that very kind introduction and for inviting me to present in this session. I'm hoping to show you some of the data that we have generated using our flow cytometry platform and how we have been able to pair that with other platforms to get some insights. And this is mainly to guide our efforts in coming up with the HIV vaccines. And yeah, so that's my title, how we are applying flow cytometry in characterizing HIV-specific B cells, and this is to guide HIV vaccine development. So our efforts to come up with an HIV vaccine are guided mainly by the poor outcome that we have had as a field in having an efficacious HIV vaccine. So here I'm just showing a list of clinical trials for HIV vaccines that the field thought were quite promising, and therefore pushed them to efficacy trials, phase 2B and phase 3 trials. And you can see that some of these were as way back as 2003, and all 12 trials did not show very convincing outputs, with the exception of 2 that had modest signals. In 2009, there was a clinical trial in Thailand that showed that 1% efficacy. And in 2021, there was a trial not of a vaccine, but of a broadly neutralizing antibody that was infused, that showed that the broadly neutralizing antibodies were able to protect against sensitive viruses. So you can see that we still have a way to go. And part of the reason why we think these vaccines failed is because they were designed using what we call an empirical approach, where you take a representative HIV virus variant, you use that to guide in designing the vaccine construct, and with the hope that if you try several, some of them will elicit a protective response. This has worked very well for other pathogens, but for HIV, because it has this very high antigenic variation, that doesn't seem to work. It always turns out that the variant that you choose to design ends up being very different from almost every other variant that is circulating out there, and so the vaccines end up not being protective. So this has triggered the field to think about rational approaches. And this is where you identify an endpoint that you think can be protective. And then you work around the pathway that can lead to that eventual output. So these endpoints that we think could be protective include things like broadly neutralizing antibodies, and that's where the B cells come in. So the reason why we think broadly neutralizing antibodies, first of all, broadly neutralizing antibodies are antibodies that have been shown to target very conserved regions in the on the HIV virus. And therefore, they are able to protect against the most of the circulating variants out there because they are targeting very conserved epitopes. And so these ones have been isolated from HIV-infected individuals, having been elicited by the natural infection in some individuals. And they have been tried in animal models, and that has consistently shown that they can protect animal models from infection if they are administered prior to exposure to the sheave in case of the animals. And actually, even in humans, one of the trials that I showed that had modest efficacy in 2021, the AMP trial, there was a broadly neutralizing antibody that was administered to humans. And it showed that, as I mentioned earlier, it can protect against sensitive HIV variants. Meaning if you have a broadly neutralizing antibody that is actually able to cover the majority of circulating HIV variants, you could protect people. So here I'm just showing one of the older studies in animal models where it showed that if you administer these BNAbs, we call them BNAbs, if you administer them, you will protect the animals from getting infected compared to the controls. So with that in mind, when you look at the right side of this slide, it means that if our target is to elicit broadly neutralizing antibodies, we can have a rational approach to try and reach that endpoint with the hope that that would be protective. And so to know that path, the field has done many studies to study the natural HIV infection process. And how the disease progresses, and how some people eventually develop broadly neutralizing antibodies. And if you look to the left, we have studied how the immune response starts with naive B cells, a pool of naive B cells, each of them targeting different antigens, some of them targeting HIV antigens, others, every other antigen or every other pathogen that the person could ever be exposed to. And these usually undergo a process of affinity maturation in the course of the HIV infection. And eventually, some of those clones, some of those lineages of B cells that are activated from the initial naive repertoire to now become memory cells to HIV and to mature over time. Some of them eventually give us what we call broadly neutralizing antibodies. So the idea has been to recapitulate this process using vaccines. And the field has therefore gone to check also what are the variants of broadly neutralizing antibodies that humans are capable of producing. And by studying people living with HIV, especially in the era before the test and treat, where there was a lot of stimulation of the immune system because people are not on ART, people are able to isolate quite a number of broadly neutralizing antibodies. And here I'm just showing quite a number of them where the potency is shown on the x-axis and the breadth or coverage is shown on the y-axis. And you can see that we have a broad range with the various breadths and various potencies. And these are the ones that the field is using as templates. And then the field has also gone ahead and even mapped on the HIV envelope spike, the one shown on the right, where do these broadly neutralizing antibodies bind? So that those epitopes can then be used to design the vaccine constructs. And so with that information, the field has then gone ahead and tried to reproduce what happens during natural HIV infection. And one of the strategies has entailed targeting specific naive B cells that have structural features that are similar to those ones of the non-broadly neutralizing antibodies. And so we call this approach the germline targeting approach, because we are targeting specific features in the germline of the individuals. And so the idea is to prime using those germline targeting immunogens and then come with rational boosters that will shepherd that immune response to eventually develop the final product that you want to develop. And so something that I would like to maybe mention is that this approach is very dependent on what is present in the naive repertoire. If someone has the structural features that are required to develop broadly neutralizing antibodies in that naive repertoire, then the chances of developing those antibodies is higher. But if someone has maybe a genetic variation that makes them not have those naive B cells that have that potential, it means that you don't have a high chance of inducing the desired response. And so we have therefore called the naive B cells that have that feature, we have called them precursors for broadly neutralizing antibodies, and so I'll use that name going forward. So as I've just said, in every population you'll have a few people, some people who might have very few of these precursors just because of probably environmental exposures or their genetic makeup, and others might have more, and the ones who have more, based on animal studies, it has been shown that they are more likely to develop the desired response. Some of them will have genetic profiles that make them not able to harness the immunogens that we expose them to induce the desired immune responses. And so to understand this in our Kenyan populations, and also with our collaborators across the continent, we have designed several cohorts that are meant to assist us to understand whether our populations have this capability to develop these desired responses. Here I'm just showing two of the cohorts in the country. And the idea is to ensure that in as much as these products are designed in the West, with the populations in the West in mind, our populations are not left behind and they can benefit from these products. And so we have established some pipelines to study those naive precursors that I've just mentioned. And flow cytometry is at the center of all this because that's where we start, because you have to sort the desired responses and the potentially desired responses, and then you couple flow cytometry with other platforms like sequencing platforms. In this case, we use single-cell sequencing because it enables us to actually study each of those naive B cells individually and look at their BNAB features if they have them. And this is just zooming a little bit into the flow cytometry setup. Some of the reagents, we have to make them in-house. we have to take recombinant HIV proteins, couple them with fluorescence labeled streptavidin to generate tetramers, and those are the ones that we use to identify the B cells that are capable of eliciting HIV-specific responses in as much as you're studying naive B cells. So they'll be very rare, and therefore that's the reason why we need this kind of reagents. And then you stain, and then you go to the flow cytometry sorter. In this case, at the University of Nairobi, we are using the BD fax melody. And then you identify the cells of interest as shown on the bottom left corner, where then you sort them. Then you take them to the sequencing platform. In this case, we are using 10x, but you can use even a plate-based approach or any other single-cell sequencing approach, where you are able to compartmentalize each of the cells into its compartment to generate cell-specific cDNA, genetic libraries downstream, sequence, and analyze. So with this kind of platform, I'm going to show you some of the recent work that we have done. Actually, this data just came out less than a month ago. And here we are evaluating one of the HIV products, HIV vaccine products that is at the very early stages. This has not yet gone into clinical trials, but what we really wanted to know is, do our Kenyan populations have a chance to generate immune responses against this product? So the idea is to study healthy individuals or individuals who are likely to, even if these HIV-infected individuals, the populations that we think could end up participating in clinical trials or eventually even benefiting from these products if they eventually get licensed. And if you remember the cartoon that I showed earlier of the HIV envelope spike, we have one epitope for broadly neutralizing antibodies called the EMPA, membrane proximal external region, that has shown to be a very reliable target for vaccines. And so we have partners that have actually developed an immunogen from this part and actually created a scaffold protein which contains a section of that EMPA region and designed it in such a way that it can elicit responses. It can activate naive B cells that look like one of the non-broadly neutralizing antibodies. And the broadly neutralizing antibody that we are using here as a template is called 10E8. So basically here we were checking whether African or Kenyan populations have naive B cells that look like 10E8 to tell us whether we have a chance of benefiting from this. And this is showing the flow cytometry gating strategy that we used to identify the B cells and then identify the antigen-specific B cells on the top left corner, bottom right corner, where we're identifying the antigen-specific B cells that are binding to this EMPA GT12, and then weeding out the ones that are binding to the scaffold protein, as shown in the last flow cytometry plot there, by using a scaffold protein that doesn't have the epitope, and therefore being able to identify the epitope-specific cells. And then after sorting, doing flow cytometry sorting, and then taking that to sequencing. And here I'm just showing some of the data from the flow cytometry component of that pipeline. And here we had the luxury of actually comparing our very early data with data that was generated by our collaborators in the US. So the first columns there are showing data collected, generated using the same product or the precursors, the previous versions of that product by our American collaborators. And the purple dots are showing the Kenyan data. This is, as I said, this data just came out 3 weeks ago, and the team is currently planning to increase this so that we can see the general variation in our populations. But as you can see, when you look at the GT12 data from the US compared to the GT12 data from Kenya, it's similar, meaning that this product has equal chances of succeeding in our populations. And so this is basically just the antigen-specific B cells from the flow cytometry binding. And the epitope-specific B cells, when you weed out the cells that are binding to the scaffold protein, so that you're only left with the proportion of cells that are binding to the actual epitope that is meant to induce the desired responses. You can see that it's very comparable to what we saw, our collaborators saw in the US when you compare the purple with the other colors. And now from the sequencing data, we go in there and look at the B cell receptors and see whether they have features that look like the features for the 10E8 prototype antibody that we are using as a template. And we saw that that is very similar. So this gives us hope for this particular product, that it could work in our Kenyan population and probably the rest of Africa. But of course, we will get to know the heterogeneity once we increase the numbers. We have used this pipeline in the past to do other similar studies looking at products that are meant to induce responses to other epitopes, like the CD4 binding site, which is another epitope for broadly neutralizing antibodies. And that work has now been completed. I'll show you a little bit of that. And also, we have also used the same platform to analyze responses to actual vaccine samples, where some of these products have been tried in a clinical setting, and we use that to determine whether these products are actually immunogenic when they're actually administered to humans. So this is showing a snippet of the data that we used to generate population-level responses on the precursors to that other epitope, the CD4 binding site epitope. And in this case, we were comparing people from high malaria and low malaria regions, because we thought that malaria is a major driver of immune profiles. In as much as we are studying HIV-specific responses, we know that malaria could modulate the immune response in many ways. So this was just a starting point, and we didn't see a difference, meaning that this product, in this case, we were looking at a product that is meant to induce responses to CD4 binding site. The product is called EO-DGT8, and meaning that it will work across the continent regardless of the malaria malaria prevalence in those regions. And this is showing some of the output from vaccine analysis. This was a clinical trial that was done in Rwanda and South Africa, but then we were part of the team that actually we had to receive these samples in Kenya to do the analysis in Kenya, so that we could see whether the immune responses were adequate that were elicited by by that same vaccine product in humans elsewhere. So therefore, acting as a centralized laboratory between the University of Nairobi and Kemri Wellcome Trust Research Program for the region. And so here, the African clinical trial is the one shown in purple, in pink, the GIS-003 clinical trial. And we are comparing it with the green and the blue, which were GIS-001 and 02 that were done in the US. And we saw very comparable results for the GIS-002 and GIS-003 that were using the same formulation of the product, and slightly lower responses for the other formulation, which was a protein formulation shown in blue. So with that, I would like to acknowledge our groups. We have a collaboration with Kemri Wellcome Trust. Of course, I'm from Kavi. We have quite a number of people involved there. We work with many other groups across the continent. Thank you. And then our funders.
Thank you very much. Okay, we will quickly move on to our next speaker. Okay, so we've been talking a lot of data analysis and all, but now we want to move. So we would want to now introduce one of the local companies that have been supporting us, not quite local. There's Excella Technologies, working in partnership with Beckman Coulter. So this morning I have the pleasure of inviting, just give me a second, sorry, it's loading up, is a brief. I'll make this brief. Okay, so Mr. Newton is from the Beckman Coulter. He's both a manager in the East African and West African region. He's been supporting us for the last,
This is inception?
Oh, yes, no, when we started, so I met him when we started the AIBBC workshops, and he's been supporting all the workshops that we've had since, for me, I started with him in 2018. So I'm really appreciate the team for really standing and working with us on this journey as we try to bring flow cytometry into Kenya and across the continent. So really, it's a pleasure to have you here. We look forward to you just sharing what you're doing. I know you do a lot of training, but there's support, in-country support that you can offer through the Beckman Coulter Hub and the Excella Limited as well. So we welcome you. Try to give you about 10 minutes.
Sure.
That'd be okay?
Sure.
Thanks.
Thank you so much, Dr. Chola, and the organizers for this wonderful workshop and conference. We are really, as Beckman Coulter, very pleased to collaborate and to work together. From inception, as you've said, up until now, we've really had a very nice collaboration, just bringing the support that we can from Beckman Coulter. And for us, really, is a privilege to be able, even as the industry, as a supplier, to be able now to reach out to all the scientists and to be able to support your work. So we don't work in a silo, we don't work alone. In all the countries where we are, in East Africa and West Africa and all of Africa and other parts of the world, we do not work directly as Beckman Coulter. So we try to identify local channel partners that we can be able to collaborate with, because as you saw from one of the presentations, service support, application support is very critical. So we as Beckman Coulter, we just don't sell the block, we just don't sell the instrument, then we walk away. We try our level best to be able to develop the local support. And in this case, in Kenya, we are working with a local company by the name Excella Biotechnologies. So I just want to acknowledge them. I know they're in our midst. Irene and Christine, if you're there, you could just stand. So Irene is our application specialist. Thank you, Irene. And for them that were in the workshop in MKU, I'm sure you had an experience with them. The workshop was very excellent. Dr. Bernard Kanui, we really worked together with you, and Professor Jesse Gitaka. Thank you for that collaboration. It was very, very successful in terms of workshop, just giving that hands-on experience. And what we did, we brought in the DX Flex technology, which is our 13-color flow cytometer for research, but then is also approved for clinical diagnosis, both clinical diagnosis for leukemia, lymphomas, and also for the research applications. So in this case, I just want to take us very briefly through some of the products, and basically what we do, we not only do flow cytometry, but we also have other range of products. So Beckman Coulter, I know most of us, sometimes it's a confusion, because we do have Beckman Coulter Diagnostics and Beckman Coulter Life Sciences. So Beckman Coulter Diagnostics is totally different. It's a sister company, but then it does have its own structures, different management, different products. Yeah, so if you're going to Beckman Coulter Diagnostics, you'll find hematology, microbiology, urinalysis, so those are the main ones. And then when you come to Beckman Coulter Life Science, now where I manage the East African and the West African markets, you're going to find some of the things that we'll be talking about today, which is flow cytometry. You'll find centrifugation. You'll find your solutions for product particle counting and characterization, and also for those customers or scientists who are having a lot of data and a lot of samples to process, and you need a lot of liquid handling. So then we come in with when you're doing a lot of pipetting, sometimes, as you know, it depends with the person who is doing the expertise of the person who is doing the pipetting itself. So then we have solutions that can help you to do automatic handling of those liquids that you'll be handling. So in this case, for biotech, we have different solutions. So we have solutions with liquid handling, as I mentioned. We also have integrated automated systems. Yeah, we have genomics reagents that can help you both from downstream to upstream. And also we have air, liquid, and particle counting solutions that will look at it. So I'll briefly look at each cadre. In this case, the liquid handling solutions, we do have the Echo. So the Echo really is one of our newest equipment when it comes to liquid handling, where it uses the Echo. It doesn't necessarily, it just uses the Echo, you know, to the sound integration for it to be able to measure very, very micro liters that we'll be able to analyze. Yeah, then of course we do have the BIOMEC series. The BIOMEC series is meant for those labs that have multiple samples that you need to be able to pipette over time. So in that regard, we sit down with you from the inception of your project. We sit down with our engineers from Germany and also from the US. We try to guide you in the process from the beginning, so that by the end of the day, when we are engineering the instrument, we customize it based on your needs. If you want to do some centrifugation, you want to do incubation during the process of your handling of the samples, we can help you to automate the system from end to end, and we can also incorporate in the centrifuges within the system. If you need to do any basic downstream PCR, they can also be incorporated on the system from the beginning, but then we have to be incorporated from the start. Yeah, then from genomics, I know there are also our colleagues here who are doing your PCR, you're doing your sequencing. So from your DNA and RNA. So we do have extraction kits, we do have isolation kits, and also for purification kits. So we assist you during the DNA extraction. We provide kits that will support you on that. Yeah, then after, of course, you've extracted your DNA, you want to isolate it. So we also have isolation kits. Yeah, then we also do have the purification kits to make sure that whatever sample you are now placing onto your the different sequencers that we'll be using, if it's next generation sequence, Ion Torrent, whichever sequencer you're using, Sanger sequencing and all that, we can help you to be able to have a pure sample that you can be able to put onto your final sequence. So reach out to us. We have very pure. We have kits that have really shown to give you a very good yield, and we can always negotiate depending on how much you're using. We can give you good pricing for the same. Then for centrifugation, I know most of us who've been in science for many years, you would appreciate Beckman Coulter as, many people know as producers of centrifuges. In most countries, when you talk about the other products that we have, most people don't know about the other products we have, they know about the centrifuges. And I usually say sometimes it's a disadvantage to us because the centrifuges that we build as Beckman Coulter, they are meant to last. I went to ILRI one day and I found they had a centrifuge that had lasted in the lab for 30 years, which they retired. They retired it at some point. I think maybe someone was just tired using it. So they retired it and then they still brought it back to the bench. And that was 30 years later. So it means for us, when we sell a centrifuge, you might not be able to do another cell in your lab. So it's good for you, but sometimes for the industry, it's a disadvantage. But of course, we are always building on that technology. So we do have the tabletops, which are very low sample sort of centrifuges. But then we've extended also to the very high end. So we do have the ultra centrifuges that can be able to, including even the tabletops, a tabletop ultra centrifuge that can be able to give you up to 1 million Gs and up to 100,000 RPMs. So that is very, very high end. If you're dealing with proteins, you're dealing with viruses, and also you're dealing with very tiny microorganisms that you would want to look at. And then we also have the AUC, which is also an analytical centrifuge. We don't have it in this market, but it's good to know that it's available if you really want to get into proteomics and you're very specific, especially with the protein analysis and studies. Yeah, then we have also the large volume centrifuges. Last year we launched the Avanti J15, which is a smaller tabletop unit, but then it has a high volume. So if you have blood samples you want to centrifuge, it does have 4 buckets, each 750 mils, so it can give you the capability of a smaller size sort of centrifuge, but then high volume. So it does up to 3 liters of sample. But then it gives you the flexibility of a smaller size sort of space. Yeah, then if you're looking for really, really floor standing and high volume centrifuges, we also have the Avanti series, which can do 6 liters if you're looking at blood sampling, and also we have the highest that does up to 9 liters. So really 9 liters of sample, that is quite something huge, but then we give you the flexibility to be able to run on the same. Yeah, then we do have the traditional Allegras, which they are the traditional tabletops that can be able to help in smaller labs, or if your budget is limited, you can be able to still be able to use that. For particle counting and characterization and cell health solutions, so these would involve labs that you would want to look at specific cells. If you're looking at cell viability, you're looking at cell counting and viability in this case. Yeah, we do have the ViCell Blue, which is really an instrument that has been revolutionized, is a digital instrument that more or less is automated. Yeah, so it can be able to give you very good data that you're looking at. And it has a big screen, the ViCell Blue, as you can see on the screen. And most labs here in Kenya, they are adopting that. They're changing now from the manual. You know, I usually say when you're looking at the microscope, it really depends on the slide. It really depends on your expertise. People look at the same slide and they see different things. But then if you have a digital and automated cell counter, it can be able to help you with the same. So we can also reach out to us if you're looking at cell counting. Then our main line under Beckman Coulter Life Science will be flow cytometry, and also that's one reason why we're here. So with the flow cytometers, we do have 2 sets. We do have flow cytometers for research flow cytometry, and we also do have flow cytometers for your clinical applications. So we do have, I'll take us through the research aspects. So the research, we are quite, as Dr. Mbuchi mentioned, we collaborated with them as well, and as you see from my last slide, and we do have very high-end flow cytometers that can really give you more flexibility in terms of the parameters and the markers that you can be able to look at in terms of your immunophenotyping and also your research applications. And in this case, our brands for research, they are the Cytoflexes, the Cytoflex family. And in addition to that, recently we've also launched a sorter, a cell sorter. So that is also something that will be coming to you if you do really want to sort specific cells. Perhaps you want to grow them, you want to culture them, you want to have more in-depth knowledge or studies on the specific markers on them. We can also be able to provide you with the same. Then for clinical flow cytometry, a few years ago in Kenya specifically, and even the region, within the East African region, you'll find most of the clinical samples. If somebody wanted to do a leukemia, a clinical pathologist would want to run a clinical sample. They had to take the samples to Lancet, and they had to take the samples to Bristol, and sometimes Bristol in the UK, or even Metropolis in India. But we came in, we partnered with some of the institutions, specifically Aga Khan, where now we've developed a center of excellence in the clinical aspect. So all the samples have been going in. But in addition to that, we've made several arrangements, and now more people are exposed to the same. So reach out also to us if you're doing clinical diagnosis or you want to do clinical research. So this is my last slide. In terms of collaboration, we've done a lot of collaboration, and this is really a very interesting slide for me. For instance, most of us are familiar with AMPATH. So AMPATH recently, we collaborated with them. We have a medical and scientific affairs department within Beckman Coulter. We sat down with AMPATH in collaboration with Indiana University, and they were able to write a grant to NIH. And last year, they were awarded $2.5 million to be able to support clinical diagnosis in Kenya. So they've started now the process, and I believe that is something that will be impactful. We have different universities we are working with. There's Burkitt's Lymphoma Fund for Africa that is also really working on Burkitt's lymphoma that we know is very endemic to the Western region. So they're doing both research and also the clinical aspect. Yeah, then there's the project that Dr. Mbuchi presented. That we also collaborated with the York University in UK, and they were able to have a multi-country project within Uganda, where they were working with Makerere University in Sudan. They were working with Khartoum University in Ethiopia. They were working with Gonda, University of Gonda. Yeah, then in Kenya, of course, they were working with Mbuchi. And those are just some of the collaborations. We can come together. If you have a project that you would want to work on, reach out to us. We can liaise with the Medical and Scientific Affairs Board, and we can help you in case you're looking for grants, in case you're looking for grant writing skills, we can also help with that. And then we put in some of these specifications to do with flow cytometry from the beginning, and then when the grant comes out, you already have people that can support you. Locally, we assure you we do have the local support, we have local engineers, we have local applications, and also we do have myself here who can be able to support. And also within, I know we have our colleagues from West Africa, so we're also collaborating quite a lot with the countries in West Africa, and specifically Ghana, Nigeria, Cote d'Ivoire, and Senegal. We have quite a number of projects that are ongoing. And in addition to that, I think ICAS, they mentioned. ICAS mentioned that they will be in Ghana, so that is also a project that we are collaborating with, and we'll be providing IDX Flex at the Center for Plant Research, and I'll be there in 2 weeks, so I'm sure we'll be able to meet and we'll be able to collaborate together. So thank you so much once again. In case you need more information, in case you need to collaborate further, you can reach out to us at the desk, then we can be able to give you more information.
Thank you very much, Nito.
Thank you.
So I know we are bad for time, but the last speaker.
Thank you, Lucy. Thank you, Nito, for taking us through what all you have. So now, the last speaker of this session, Dr. Kennedy, and it's my privilege to introduce him to all of you. I mean, he doesn't need any introduction, as you know. He's the man running around, full of energy. Okay, but then also I'd like to formally introduce him to all of you. He's a technical information scientist and a product manager, works at JAX, which is the Jackson Laboratories in Japan. Plus, he's also an adjunct faculty at Purdue University, USA. In the past, he works as a senior lecturer and assistant professor in Tokyo. And he's contributing a lot to African science and arranging these meetings and the workshops in collaboration with Kenyan scientists here. So I can say in one line that he's giving back to its roots, which is really appreciable. Thank you, my dear friend, for all your work that you are doing for Kenya. Thank you.
Thank you. Thank you. Oh, my God. Thank you, Hammond, for such a flowery introduction. I'm so humble. Now, the one standing between you and your lunch, and I must apologize in advance. This is going to be an elevator talk, so I will be very fast through the slides. I'm given about 5 minutes to clear my presentation, so please bear with me. I'll be running. Okay, just wait. Oh, thank you, it's here. So today I would like to talk about something which may be a little bit different from what others have talked about, but I think the foundation has been laid. So I will just go through what we are doing toward profiling human immune cell engraftment in humanized mouse models. Perhaps it's a little bit different, and I may have to go through the basics, but I will be kind of moving very quickly, so please bear with me. So just a brief introduction about JAX. JAX is an independent, nonprofit biomedical research institution. Our mission is to discover precise genomic solutions for diseases and also to empower global biomedical community. So we do provide animal models, but my role is to oversee preclinical testing using some of the various models that we develop. So I will precisely talk about some of them here and to tell you what we do also with flow cytometers. So as we know, cancer is a big problem, and also it's an emerging challenge here in our continent, but there are many various factors that we have to look at when we think about cancer. We have hematological cancers, liquid cancers, we have solid cancers, but the biggest challenge that we see in the field is with regard to the solid cancers. And the key thing is the tumor microenvironment, which is a very complex ecosystem consisting of immune cells, the cancer cells, fibroblasts, and several others. So the strategy, several of them are being developed to actually address the challenge that we face with solid tumors. And a lot of modalities, we see the pharmaceutical companies and many other bioventures coming up with different aspects of drugs that do target tumor cells. They comprise about 80% of all the cancers that are being seen in the clinic. So our role is to develop preclinical models that assist in the testing of the modalities being developed toward cancer treatment. And there are so many 2D cultures, organoids, 3D cultures, organ on a chip, but today I will focus more on animal models. So just to run through, like for instance, one of the modalities that we do test a lot is CAR-T, which is the chimera antigen receptor T cells. I will not go into the details because perhaps you're more informed about this than I do. But the CAR-Ts, we take them from the patients. We use retroviral transduction to induce the antigens on the T cells, and then they are taken back to the patient. So we do test these kind of modalities before they are taken to the patient, just to see whether they act against the tumors that they are supposed to address. So animal models are very important in preclinical testing of all the drugs that we see in the clinic. And our role is to make sure that before we test them in humans, the drugs are very safe, their efficacy is proved. And so some of the models that we do develop, CDX is primarily, we use the cancer cell lines and we transplant them into the mouse, and then we develop the cancer, and then now we can test other modalities. And another one is we take tumor from the patient and we directly transplant those tumors into the mice, and then we use them to test drugs and other modalities that we develop. So one aspect that I want to introduce today is a special kind of mice that contain the human immune system. So these are called humanized mice, and we do develop them because we need the immune system to be present when you test modalities against cancer. Previously, our existing models, we have different kinds of models, immunodeficient mice, but they lack the immune system, meaning the interaction between the cancer and the drug that you develop will be missing if you test them in just normal mice. So we developed these special kind of mice that contain the human immune system. So the starting point is the immunodeficient mouse. It doesn't contain its own immune system, so these are special kind of mice. And then in the mice, we transplant PBMC, and sometimes we do CD34 human hematopoietic stem cells. And when we transplant them with either PBMC or the CD34 cells, inside the mice, we develop the immune system. So the mouse contain human immune cell population, DNA, RNA proteins. They express cytokines and growth factors, and also they can allow you to now decipher the human cell-cell interaction between tumor cells and immune cells. We do use them to carry out evaluation of very many different aspects of cancer modalities. For instance, we use immune checkpoint inhibitors, CAR-T, BITE. I will not go into details of this. Please, you can check more information on your own. So the way we do this, just as I've explained, we take, for instance, the human, we have the leukoplaques. We just take PBMCs, we inject them in the mice, and then after about 2 to 3 weeks, we develop these mice containing human immune cells. And also CD34, it takes about 3 to 4 weeks to develop, but then after about 3 months, it takes a bit of time to have the full immune components developing in the mouse, and then we get a variety of immune cells in the mouse. I will skip the humanization scheme due to lack of time, so I will just quickly go to some of the data that we have. We have developed quite a range of platforms for doing this, and some of the mice here, they express IL-15, which is very important for the survival of NK cells. We have SGM3 expressing mice, and all these are developed to support the human immune system development. I will focus more today on the NK cells and to show you a little bit how we can test drugs and other things using these kind of mice models. So this is the whole developmental process of immune system. It starts from hematopoietic stem cells, extending to lymphoid, progenitor cells, and various other kinds of cells. But I want to focus more, on the, how do you get back? Okay, so the NK cell actually requires IL-15. It's very critical for the survival of the cells. So what we did is to knock in IL-15 into the mice, and then now they can sustain NK cells when we inject them with PBMC or the cord blood-derived CD34 cells. This is the scheme that we used, and we selected about 2 strains of mice plus others. So what we do typically is after every 2 weeks, after injecting them with CD34 cells, the immune system develops. So we have to really characterize what sort of immune cells develop at what time point. So we do carry out, we have a panel of CD45, CD3, CD20, CD33, quite a range of panel that we design, and we use a flow cytometry to analyze the development of immune system in these mice. So for today, I will just quickly go through and just tell you how we look at the dynamics of immune cell development inside the system. You will see, for instance, here, this is CD4 in blood, spleen, and the bone marrow. And you will see that as days go by, we can trace them for a couple of weeks, even 28 weeks, which is a very long period of time. We can start from week 4, from week 1, week 4, all the way to week 28. And you will see the kinetics here. We have the numbers of immune cells will keep on going up as days go by. And so that means the immune system is developing inside the mice. And also you will see the CD8, the numbers will be going up, but then we have a peak at some level. It peaks and then it becomes stable and can be sustained for several weeks. I just want to skip, run over this. And then we can also capture T regulatory cells, which is a very important component of the immune system, and just won a Nobel Prize this year. So we'll see they can also develop in these kind of mice, and they will be going up and stabilize at some point. And then you can see that in the spleen. Primarily, some mice do not have them, so we have to knock in some cytokines, growth factors that sustain the immune system in the mice so that we can sustain the cells. I will just go quickly and tell you a little bit about mature NK cells, very important when we consider new modalities targeting NK cells. And you will see we can also sustain, we can get high levels of NK cells in specific mice expressing IL-15, which is very important for the survival of the cells. But in bone marrow, we don't really see NK cells developing. So it is primarily in the blood and also in the spleen, we see a number of NK cells there. So I jump to the preclinical applications. This is our system. I will just once again highlight that we inject CD34 hematopoietic stem cells. We have the humanization taking place. And then now we can take a tissue, a cancer tissue from the patient, implant them in the same mouse, and then we can now test modalities such as anti-PD-1 cancer immunotherapeutics. So a quick review, we have to test whether the NK cells developing in the mice, whether they are really functional. We do see them expressing granzyme B, and also we see spleen and blood, they do express those granzyme B, so it means that they are functional. We can do in vitro studies to evaluate how their functionality compares with PBMC, and we see that the comparison is very nice. You see the black line is human PBMC. The NK cells from human PBMC compares very well with the NK cells that develop inside the humanized mice. And now I want just to clarify one drug that we did test, which is the ADCC. And what is ADCC? This is just an antibody binds on the surface of a target, which is a cancer cell, and then NK cells now will recognize the antibody, and then they destroy the cancer cell. So this is called antibody-dependent cellular cytotoxicity. It's one of the models that we now see being developed by big pharmas to target cancer cells in many different cases, especially solid tumors. So this is a study that we did to just verify if our model can actually recapitulate what we see in the clinic with ADCC. So we chose on this tumor cell line, which is very resistant to many NK cell kill. They are not really targeted by normal NK cells. So we wanted to see if this drug, rituximab, is a drug. I've written down the specifics there. But just to show you that in the mice, if we have the normal PBS as a control and we have the rituximab as the drug, you will see the green lines. The mouse, some mice respond, some mice don't respond. So we capture what we call the variability that we see in the clinic. And also you will see this, the drug can be tested in the mice, and you will see that the green line on the left, we have the efficacy is actually going down. We see that the drug is actually working in the mice, and we can see this, the tumor volume is actually sustained, is very suppressed in the case of the drug treatment. I will just want to skip the slides and end very quickly. This is just showing you that we can go deeper using flow cytometry to see how the drug is acting. So the most important thing in cancer now, in solid tumor, is to see whether our immune system is penetrating deeper into the tumors and trying to kill the cancer cells. So in this case, we do what we call the tail analysis. So we take the tumor, we break it open, and then we run a flow cytometry to see how many human immune system penetrated deeper into the tumor. So this is showing you that in the case of this treatment, I will try to skip the specifics, but you will see the CD3, CD8 actually are infiltrating into the tumors, and then when you have this drug treatment versus the control, you will see that we have a better penetration with the drug that stimulates your immune system now to go and target tumors. I will not skip this one because of lack of time, but this is just a flow cytometry analysis at the end point, and we do see in the blood, spleen, and tumor, we can do separate tissues of a patient, or sometimes we also do this in the mice, and we do comparison between what we see in the patient and what we see in the mice, just to test that the models actually work, whether we have it in the mice or we have it in patient. The key role here is whatever modality that we test, whether it's a drug, it should work the same way in the mice as well as in the patient. So we want to do this before the drug is given to you. The mouse will tell you whether it will work for you or it will not work for you. So this is what we do to verify efficacy before a drug is developed and it goes to the clinic. So just to summarize, we have developed a couple of preclinical mouse models, and we are using them to advance treatment of different cancers through testing their efficacy and safety in the models before they go to the clinic. And also we are trying to recapitulate the human immune system, including the NK cells that I've talked about today. And we have seen that the NK cells are mature and functional, and we have seen application in ADCC-based cancer immunotherapeutics. So with that, I would like to end, and I think it has ended. So any questions we can discuss over lunch, and thank you very much.
So thank you, Dr. Kennedy, for such a informative lecture that you gave, and I think everyone got benefited from this. So as we don't have a time for the question, but we will welcome that you can interact with Dr. Kennedy over the lunch. So now we all break for lunch, and at what time we have to?
We'll be back at 2 o'clock, is that right? What time is it now? It's 1:30. Let's try as much as possible to be back at 2. I mean, I'm not going to give you, I might be lenient and maybe 15 minutes, but I'll push you to come back at 2. As per yesterday, we have given different vouchers for lunch. So if you have the purple one, you're going to the 10th floor. And if you have the green one, you're going to the 3rd floor. And again, I assure you there's no difference in food. It's just because we have a large number of people. And in case you don't have a voucher, I think Joy should be outside. You can see her, and she'll hand one over to you. Bon appetit.